Right, so I know that maybe for weather some people still are coming, but I think we have to get started because actually I'm gonna start.
Yes.
Yeah, so otherwise we cannot finish everything. So all right, good morning everyone. So how is everyone doing today? Good? Tired? Exhausted? Okay, so today we are going to actually cover a very exciting topic. Okay, microbial electrochemical systems for waste and wastewater treatment. So far over last 3 days we kind of focused on more on conventional bioprocesses for waste and wastewater treatment, and today we are going to see a very emerging process. Okay, microbial electrochemical technologies. We are going to see like how we can actually use this process for waste and wastewater treatment. So anyone here familiar with microbial electrochemical systems before? No? So microbial electrochemical systems, actually that is an environmental bioprocess. Okay, so in conventional environmental bioprocess, what we do, we utilize microbial metabolism, right? So here, in addition to microbial metabolism, actually we add electrochemistry there. Okay, so we combine microbial metabolism with electrochemistry. So basically, when we talk about electrochemistry, it's electrochemical cell, right? That we can add. So basically, like an electrochemical cell with anode and cathode, and you can have oxidation reduction reaction. So for example, you can have on anode you can have oxidation, on the cathode you can have reduction reaction. And but one of the unique feature of microbial electrochemical system is here we use a unique type of bacteria. Okay, so those bacteria, they are electrochemically active. We know that actually like all microbes, they transfer electron, right? Like we learned, they actually kind of transfer electron from electron donor to electron acceptor, right? So there is electron transfer there. But this unique type of microbe, electroactive microbe, their respiration process unique in a sense that, okay, they can, so like for example activated sludge process, you provided oxygen as electron acceptor, right? But here they can actually transfer the electron to electrode surface. Okay, so that means some insoluble conductive or solid material, okay, they can use as electron acceptor. Okay, so that's the unique feature of microbial electrochemical technologies. But actually there are many terms actually over last 20 or 25 years researchers have used many different terms. Okay, and sometimes actually it's very confusing for someone actually start working in this area for first time. So for example, like there are many different application of microbial electrochemical systems. So for example, microbial fuel cell you can actually produce electricity, like like a regular electrochemical or regular fuel cell. You can actually produce microbial fuel cell can produce electricity using electroactive bacteria. So we call it MFC. We can actually have we can apply some voltage, okay, and we can get a wide variety of product like including biohydrogen from microbial electrochemical system. We call it microbial electrolysis cell, MAC. Okay, MEC. Then some researcher found that, okay, we can actually apply this concept for desalination of water. Now people call this microbial desalination cell, MDC. Okay, then researcher found actually we can actually produce different types of value-added product on the cathode side, okay, using carbon dioxide as carbon source, and that process is called microbial electrosynthesis, MES. So now lot of new application. Okay, so that's why then researcher thought, okay, we kind of give a common name. Okay, we call it MXC. So that X may represent different application. It could be fuel cell, it could be electrolysis cell, it could be desalination cell. Like then actually researchers, they have shown that you can do denitrification using this concept. So now MDC for microbial desalination cell, MDC for microbial denitrification cell, right? So there are a lot of overlap, many names, then we kind of end up with using MXC, okay? And any technology that is based on this concept, we call it MAT, microbial electrochemical technologies. Sometimes we also use the term called bioelectrochemical cell or bioelectrochemical systems, okay? So how that works, okay, like any traditional environmental bioprocess, we have oxidation reduction reaction, okay, but here that thing actually happen on the electrode, okay. So what happened, like when actually we provide those microbes EAB, so we have anode and cathode electrode and two chamber. Sometimes depending on the application, anode and cathode chamber can be separated by membrane, and typically those electroactive bacteria, they produce actually biofilm, they kind of form biofilm on the anode surface. So when you actually provide them with electron donor, they can actually oxidize those electron donor and transfer that electron to the anode surface, okay, anode surface. And the process actually also produce carbon dioxide and proton, and then it kind of transfer electron to the anode surface. Then actually because of potential gradient, that electron can transfer from the anode to the cathode, okay. And cathode, you can manipulate the terminal electron acceptor, right? So like for example, like activated sludge process, you provided oxygen and oxygen was reduced to water, right? So depending on the application, we can manipulate like what we want from the microbial electrochemical system. We can manipulate the terminal electron acceptor here and can get different types of value added product, okay. So when actually electron transfer from anode to cathode, to maintain charge neutrality, the proton that is actually released from the oxidation of your electron donor, proton kind of also transfer from anode to the cathode, because one negative charge is going there, so one positive charge should also go there to maintain the neutrality, right? So and going back, that electroactive bacteria, okay, they also actually researchers, they also gave them different name, okay. So some people call them electroactive bacteria, EAB. Some researchers actually name them anode respiring bacteria, because they can do respiration on the anode surface, okay. So they give them a name in anode respiring bacteria, ARB. Some researchers call them exoelectrogen, okay. So why exoelectrogen? Like like you remember the name from anaerobic digestion, methanogen, right? So microbiologists, they kind of like to actually name microbes, okay, like some sort of like gen, right? So those microbes, they can do extracellular electron transfer, so that's why researchers name them exoelectrogen. So again, there are many different names. So couple of years ago, the research community, they agreed that okay, what, like it's better to call them electroactive bacteria, because now we know that actually they can be not only just anode, they can be in other engineered system as well, okay. Sometimes even they can be on the cathode as well. So maybe it's better to call them like electroactive bacteria or EAB, okay. So even when I back in like between 2012 and 2015, when I actually did my PhD, I also used to use the term like anode respiring bacteria at that time, but now we call it EAB, electroactive bacteria. So here on the cathode, you can manipulate the terminal electron acceptor and you can get different type of product. So for example, like in so there are actually oxidation on anode, and cathode you have reduction. So for example, if we provide acetate as substrate, okay, acetate as substrate for or electron donor for microbes, you can see this half reaction. Like in very first lecture, we learned about half reaction, right? So if acetate is oxidized, it's gonna release carbon dioxide, proton and electron, you can see here, right? So this is oxidation. So in cathode, if I just provide oxygen as electron acceptor, then oxygen gonna be reduced to water. Okay. So we can actually, so this is kind of like the basic reaction, but we can have you know, like different electron acceptor on the cathode side, and we can have different product that we want. So this kind of electroactive bacteria was actually first found in natural systems. So one researcher actually, he is a microbiologist, okay, in US. So his research team actually found this kind of microbes in sediment, river sediment in US, one river in US, and they found these kind of microbes, okay, called Geobacter species, have some unique feature. So they can actually oxidize ferric to ferrous iron, okay, using extracellular electron transfer. Okay, so they can actually convert ferric to ferrous ion, and then, and at the time actually people didn't know that actually they can also work as a electroactive bacteria and microbial fuel cell. But when microbial fuel cell research became very popular and people start to actually do microbial analysis and try to know, okay, what kind of microbes are there, they found that actually, you know, in most cases Geobacter species is a common type of microbe. Most of the time we find on anode surface of microbial electrochemical cell. So EAB, they can respire, so they can do respiration. So this is actually a unique example of anaerobic respiration. Okay, so anaerobic respiration, and because they can use insoluble electron acceptor like metal, minerals, like, you know, in this example they used metal, right? But in our case, it's when we talk about microbial electrochemical cell, they are using electrode, right? They are actually transferring the electron to electrode. So, and EAB, they use unique electron transfer mechanism which is called extracellular electron transfer. Okay, so they have, like, you know, they oxidize the organic compound and transfer that electron to their outer membrane electron transport system, okay, like other microbes, but then that electron is actually transferred to the actually electrode surface, right? And that part is called extracellular electron transfer, or EET. Okay, so because of this feature, okay, they are kind of considered as unique. So in addition to Geobacter, other common type is Schwannella species. They are another type of bacteria that can do is actually this kind of EET. But later on, there are many other bacteria have been discovered, okay, they can do EET, but we, you know, like maybe 10 or 15 years ago, it was not known that actually those microbes can also do EET. Okay.
So these are, we can say, anaerobic bacteria?
Yeah, they are anaerobic bacteria, yes. So they don't need oxygen, right? So they, without oxygen, they can do their respiration. So they are anaerobic bacteria. So here you can see here, like, you know, there are three major steps involved. Okay, three major steps involved, like, you know, electron transfer from electron donor to the electrode surface. So for example, you know, first step, microbe, they oxidize the electron donor, release electron, and then intracellular electron carrier, like, you know, NAD plus NADH. Okay, this is a redox cofactor, or can act as electron carrier. So they transfer electron to the outer membrane protein, which is kind of microbial electron transport chain system, okay, or we call it ETS. And then from outer membrane protein, okay, electron is transferring from the outer membrane protein to the electron surface, which is called EET. Okay, EET. So that part is kind of like the unique feature of this kind of microbe, because sometimes what happen, a microbe, they can be very far from the electrode, okay, but they can still transfer electron to the electrode surface. Let's say, you know, like you have what is the size of microbes? Few micrometer, okay, but you can have 100 micrometer biofilm. Okay, biofilm thickness could be 100 micrometer, but those microbes still can oxidize electron donor and transfer electron to the electrode surface, like long distance electron transfer. So that is called EET. Okay, so that's why it's so unique. So how microbes they do EET? Okay, so not a single mechanism. Microbes they can use many different mechanism, okay, or combination of multiple mechanism. So first researcher actually thought maybe like those microbes when they are actually form biofilm on the electrode surface. So there is some direct contact with the electrode and they can actually transfer electron. So for example, I would say like if we draw this diagram, so researcher first thought okay, microbes they are going to be in direct contact with the electrode and they can actually transfer electron. But now most of the time researcher found actually like monolayer. So this is kind of monolayer biofilm, okay, monolayer biofilm. You have a single layer of microbes. But researcher found okay, you know what, like when we have multi-layer biofilm, actually multi-layer biofilm can produce even more electricity. So then they thought okay, you know what, that means those microbes they are actually outer layer of the biofilm, they can do long distance electron transfer. So they are not necessarily those microbes need to be actually attached to the electrode surface, right? So then they decided that direct contact is not enough to produce high current density from this kind of system. So other mechanism might be active there. Then researcher found actually microbes they can use some soluble redox material, okay. So redox active material. So what is redox active material? They can be either oxidized or reduced, okay. So we call them actually redox mediator or shuttling compound. So how that works? Actually, so let's say you have some electrode, okay, and you have some microbes in suspension, okay. Now if you have this kind of redox mediator in your system, what's going to happen? So those are redox active compound. They can go to the outer membrane protein, okay, outer membrane protein of microbes, and they can be reduced. They can take the electron from the microbe and can go to the electrode surface and can be oxidized. So from oxidation they are going to release the electron and then they go back again and then reduced again, come to the electrode surface, then go back and forth, okay. But those are soluble compound. So think about if you have a continuous reactor, okay, continuously you are actually like feeding that with wastewater, those compound may be washed out from the system, okay. So some researcher they did experiment, they added this kind of material, okay, in the reactor, and then they found actually current can be increased in batch mode, but when they actually operate in continuous mode, okay, it's a kind of like a don't show high current density. So then researcher realized okay, that mechanism may not actually lead to high current density from this kind of system. Then in actually I think it was in 2007 or 2008 researcher found actually, or maybe 2005, okay, almost 20 years ago, researcher found that actually those microbes can actually produce hair like appendages, protein appendages, okay. So they can actually produce some wire and make a connection with the electrode surface. So even they are outer layer of the biofilm, they can actually produce some wire, okay, and use that wire to actually transfer microbes, and those are actually like produced by microbes. So sometimes we call them nanowire produced by microbes, or some researcher actually call them pili, so semiconductive pili, and researcher actually confirmed that those hair like appendages actually they are semiconductive in nature. And then another actually mechanism have been proposed. So what is that mechanism? It's called electron hopping. So what is electron hopping? Electron is jumping from one hole to another hole, right? So what they actually proposed, that there could be, instead of this kind of soluble compound, okay, there could be some compound in the biofilm, they are also redox active and they are very close to each other. So what happened, they can be, you know, like oxidized and reduced. There could be a chain reaction of oxidation reduction, and ultimately electron can reach the electrode surface. Okay. I know that what I explained may be still not clear, so let's see some figure. So first mechanism, direct contact. So microbes gonna be in direct contact with the electrode surface, but we know that actually that cannot produce high current density. Redox mediator or shuttling compound can be another option, okay, but they are soluble, so they can be washed out from the system. They can be either oxidized or reduced. So on the outer membrane protein, they are going to be oxidized or reduced.
Outer membrane protein, oxidized.
They are going to be reduced. They are going to take electron,
Yeah.
right? And then on the electrode surface, they are going to be oxidized. Okay. So and going back, they are going to be reduced again and oxidized. So they are redox active compound. Okay. So they can be either oxidized or reduced if needed.
Give some example of this.
It's kind of, I forgot the name, but researcher found that some microbes can produce this kind of compound, but I can get back to you. Maybe tomorrow I can give you some name. Okay.
Do you think NAD plus could be such compound?
No, it's NAD plus NADH. Actually, this is intracellular compound, not extracellular, right? So
Can you confirm or know that is pH has some kind of relation with this kind of mediator?
pH, okay, this kind of microbes, they prefer neutral pH. So you know, like at low pH, if some of this kind of compound even active, microbes not going to have any activity. Okay.
That could be another challenge in wastewater treatment.
Yeah. So pH has to be close to neutral pH. Okay. So those microbes are actually very sensitive about pH. But now you can see, like the microbial nanowire, those microbes, even they are actually far from the electrode, they can produce those hair like appendages and transfer electron. And then another mechanism is when they are actually far from the electrode, they can also use some redox cofactor. Okay. So redox cofactor, so they are actually like mediator, but they are immobilized in the system, immobilized within the biofilm, and they are very close to each other. So electron can be actually, you can have a chain reaction of oxidation reduction. Although here you can see those cofactors are very far, right? So I have one cofactor here, one cofactor here, but in practically they can be within like nanometer distance. Okay. So nanometer distance, so they can actually exchange electron.
So how they use this pili for electron transfer? Because as much as I know, these are sex pili. The bacteria are using these pili for conjugation.
It's yes, but this is actually a little bit different. Those are not yeah, like the researcher found actually their I think structure is quite different, you know, like quite different, and they are not actually found in all my bacteria. Okay. So the mechanism C and D, so microbial nanowire and electron hopping, we consider this method as conduction based DET. So microbes, that biofilm kind of, it's kind of act like a semiconductive material. Okay. So some researchers also call it not semiconductor, they call it metal like conduction. Okay. But it's actually, it has been found that microbes, they can form a biofilm. Okay. And that is also kind of semiconductive in nature. So I am not sure whether you can see here, but you can see, like, you know, let me see if I can zoom in. Do you see it now? You can see some lines, right? So those are actually pili. So this is microscopic image of pili. So microbes, this is actually produced by Joyabrata. Okay. And then even Schwannella could also produce. You can see those Schwannella microbes, actually they also produce this kind of pili. Okay. And they are mostly protein. So then what researcher did, they separated, okay, they extracted those pili from the microbes, okay, and then what they did, they used actually atomic force microscopy and they applied some voltage, okay. So you know Ohm's law, if you apply some voltage, you should, there are gonna be some resistance, okay, and you should see current, okay. So if you, so you can see here they applied different voltage and then they found actually a very, like, you know, ideal ohmic response. So with increasing in voltage, current is increasing linearly. So this is actually like ohmic response. So that means it's kind of like, you know, those material, they are actually conductive. So this is kind of actually confirmed that microbes, they can produce conductive nanowire. Actually the same thing was done for Schwannella, okay. So I'm not sure whether you can see it. So they have 2 electrodes. So those are 2 electrodes, and then there is a like those nanowire that is actually extracted from Schwannella species. I'm not sure whether, let me zoom in. Now do you see a line? So this is actually a pili. So then they actually applied voltage. Then they applied voltage, okay, and then again they found actually almost similar pattern, okay. So with increase in voltage, your current is increasing, right. So that is actually confirmed that like Geovector species, Schwannella species can also produce conductive nanowire. But the difference between Geovector and Schwannella, actually researcher found that some Schwannella species, okay, they don't have actually so mutant deficient in gene, they don't have any gene for C-type cytochrome, okay. So they don't have any gene for C-type cytochrome. If they try with this kind of pili or nanowire from Schwannella species, you can see with increase in voltage and current line is flat. So for them actually, you know, like it's not showing any ohmic response. So that means it's not conductive. So then researcher actually found that actually for Schwannella species, C-type cytochrome would actually be the key factor in extracellular electron transfer, okay. So then researcher actually proposed the method ohmic, you know, like electron hopping. So what is electron hopping? So microbes, they are going to still produce nanowire, okay, but that nanowire contain lot of cytochrome C, okay, and they can actually kind of work as a like hole for electron, okay. So when they are, and as I said, like, you know, they gonna be, although it look very distant from each other, but in reality they could be in 1 to 2 nanometer distance, and then actually electron from outer membrane protein can actually jump from one cytochrome C to another cytochrome C, okay. So although I'm saying it's jumping, like, you know, because it's, we are calling electron hopping, but in reality it's oxidation reduction reaction, okay.
So these cytochromes are produced by bacteria?
Yeah, they are cytochromes are produced by bacteria. So for Geovector, so if I summarize, Geovector, the whole nanowire itself, it's like an electrical wire, okay. But for Schwannella species, it's it look like electrical wire, but it's not. Mainly it's actually the work is done by cytochrome, okay. Then another researcher, he proposed another mechanism. So he said, okay, you know what, sometimes some microbes, they don't produce, okay, they don't produce actually nanowire, but still they can do long distance electron transfer. So that means they actually use some actually redox cofactor, they are immobilized within the biofilm, okay. So some when the microbes, they form biofilm, they can produce EPS, right. So they produce EPS, extracellular polymeric substances, that can actually help the biofilm formation. So some of those EPS material can act like redox cofactor as well, and that can actually also help you to transfer electron from outer membrane protein to the electron surface. So that is actually another mechanism of hopping. When microbes, they don't produce nanowire.
Sir,
Yeah.
actually, you talk about the responsibility, and there is genes for C-type cytochrome, and they are producing this C-type some protein from this C-type cytochrome, and I'm asking a question that sir, this C-type cytochrome is ubiquitous in plants too, but what are the difference between them and this type of microbes that they are showing conductivity, not any other plant? Why they are so specialized?
Okay, so if I understand your question correctly, okay, why this kind of microbes are unique?
Unique, because the cytochrome C is ubiquitous.
Okay.
So, what are the specialty that this type of microbes have?
That question I cannot answer. Why? Now I have to go back and see, who created bacteria and why they were actually provided with some unique feature. Okay, I can't really tell. Okay, but there might be actually, let's say, in natural environment, there might be some situation where those microbes, okay, they did not have, they were in some environment where they did not have enough electron acceptor. So they kind of, somehow to survive, okay, change their metabolic pathways. Okay. So that could be one of the reason, but honestly speaking, I cannot answer why those microbes have that.
Actually, I'm asking because I have some organism which shows this kind of property with another element. This is heavy metal. So this is a plant. So that is why it is also having this C type of cytochrome.
Mm-hmm.
But I don't know exactly what is the mechanism behind it. So when you say that there is reason behind is the genes for C type cytochrome, I thought the same mechanism could be applied for my organism too.
So did you do any microbial analysis?
No, not
Okay, so maybe it's better to do some sequencing, okay, and you can see it's mixed culture, right, or pure culture.
just that.
So pure culture, you should know what kind of microbes.
No, it's actually plant, not microbes.
Okay. Honestly speaking, I cannot really answer that, but we can discuss that more, after the lecture, okay, if needed. So I'll move on. So microbes, they need some sort of conductive pathway for EET. So now then researcher thought, okay, you know what, now if conductive pathway is important, so the biofilm itself should be conductive as well. Okay. So now researcher tried if we can prove that actually biofilm is conductive. Okay. So how we can do that? Okay. So that was actually very smart approach. Okay. Published, I think this paper was published in about 2010 or something, this method was proposed. So you can have two electrode, okay, with some very small non-conductive gap. So they are actually two electrode on a glass base, okay, and there is a small non-conductive gap. So that non-conductive gap could be like 10 micrometer or 25 micrometer. So microbes, okay, so what is the size of microbes? Let's say 4 or 5 micrometer, right? So that non-conductive gap is about let's say 25 to 50 micrometer. But when microbes form biofilm, okay, so they can actually cover that non-conductive gap as well. Okay. So now, so here although it look like one electrode, but you have two electrodes. So what you can do using a source meter, okay, you can apply some very small voltage, and if that biofilm is conductive, then electron should be able to, because there is two electrode, they are not connected, right? The only connection they have is the biofilm. They are actually like connected by biofilm, not by any conductive material, right? So what I am saying here, So you have two electrode. So let's say electrode A and electrode B, okay, and over time you have biofilm, they kind of grow. So and those A and B, they are not connected, right? There is no short circuit in there, and now they are actually, you have over time you have biofilm formation, okay, and you have the biofilm. When they form biofilm, you have BPS and other stuff that can actually cover that gap, okay. So now those two electrodes, they are only connected by biofilm. Now if you apply some voltage, okay, across A and B, if they are actually, the biofilm is conductive, you should see a ohmic response. Biofilm should actually act like an wire, okay, and then actually researcher confirmed that. They applied small voltage and they actually found like pili, they also found like, you know, very perfect ohmic response, okay. So voltage and current, okay. So now if you actually, that kind of confirm that if you see an ohmic response, that confirm that actually like biofilm is conductive. So if you know the biofilm thickness, and from you know like this response using Ohm's law, you can actually estimate conductance, and based on that, using an equation, you can actually you know like calculate conductivity, which is, let's say, millisiemens or microsiemens per meter, okay. And then the researcher group, actually it was, you can see all of them are published by one researcher, okay. So they tried different actually strain of geovector species, okay, and they found that actually for different geovector species, actually current density ampere per meter square actually show a perfect linear relationship with biofilm conductivity. So that means biofilm conductivity is very important to get high current density from microbial electrochemical system, okay. So conduction is the dominant pathway for high current density in microbial electrochemical system. So now before I jump into the next topic, so next topic, actually now we talked about fundamental, how that thing work, okay. Now we are going to talk about little bit of application, what are the different application, and then the third topic gonna be, majority of the portion gonna focus on microbial kinetics, and we are going to learn, like we did some we learned microbial kinetics on day 2 for activated sludge process, right. And I was telling you that if we learn the kinetics for one biological process, we can actually learn other biological process very easily, and you are going to see today actually how we are going to use like almost similar kinetic equation, and we are going to come up with an equation that can allow us to estimate current density from microbial fuel cell, okay. So but before I jump into this topic, any question? Yes.
This process can occur in all types of cells which have external electron acceptor, like you showed the example.
All types of external electron acceptor. Well, it depends, okay, what kind of process that you are looking into. Some of them might be thermodynamically possible, some of them may not be thermodynamically possible, okay.
I believe that electron acceptor is not the only necessity. It should be redox. Actually, it could handle redox reaction. Then only it can serve our purpose. Right?
So yes, sometimes, like you know, I actually have some slide like explaining this, like you know, thermodynamic significance there. So let's say you may consider, okay, you know what, I'm going to put that electron acceptor there, and this is the product that I want from this reaction. But the question is whether it's thermodynamically feasible or not. You have a redox potential for your donor, right? You have a redox potential for your reduction reaction, right? But now from this redox potential, you can tell whether it's thermodynamically possible or not. If not possible, that means you have to provide some energy from outside to overcome that thermodynamic barrier, okay?
Sir, but it should be possible for the survival of the cell.
Yeah, then yeah, if they don't have electron terminal electron acceptor, yes, microbes gonna actually.
He's talking about the external electron acceptor, not internal. I guess you are explaining about internal electron acceptor.
So you are talking about external. Yeah, he's talking about external.
But you said about
I'm also talking about external. On cathode,
how
let's say.
microbe will die? How microbe will die if they do not have external electron acceptor?
So who is they have to transfer this electron to somewhere,
Proton pumps will stop working.
right? So after so for example, like I give you food, but I don't allow you to breathe oxygen, then you are gonna die, right? The same, it's as simple as this. But okay, so let's discuss, let's go through the slide, okay, and I'm going to explain the thermodynamic significance, and then we'll come back to your question and see whether it's clear or not, and we can have more discussion, okay. So we can actually, so we learned so far that actually we can have electroactive bacteria on our anode, okay, biofilm, and we can actually provide them with electron donor, and we know that waste and wastewater, they are very good electron donor, okay. You have a wide variety of organics, and we can actually get different types of value-added product if we can manipulate the terminal electron acceptor. So the question is how we can actually get different types of value-added product. So the first application of this technology was microbial fuel cell. So what is microbial fuel cell? You have anode biofilm, they oxidize the substrate or electron donor, release electron and transfer electron to the electrode surface. Electron gonna go to the cathode, and you have oxygen, and cathode oxygen reduction happen, and then you have an electron flow, which is electricity, right? So you can have electricity that you can use. So this is the basic application. Then researcher thought, okay, you know what, electricity, sometimes people consider, okay, this is cheap, right? We want to get more high value from our waste or wastewater. Then researcher thought, okay, what about producing hydrogen instead of electricity? Okay, so what if we kind of use the electron and convert that into hydrogen? So how we can produce hydrogen on cathode? So we can have proton and we can have electron that is actually coming from anodic oxidation, and then we can have this half reaction. So proton and electron can be combined together and you can have hydrogen production. But for this process we need to supply energy from outside. And why is that? Because it's thermodynamically not possible. So typical applied voltage is about more than 0.25 volt, but you can see now you can say, okay, you know what, if I provide energy, why I don't just do water electrolysis, right? Why I should do microbial electrolysis, right? But if you compare with water electrolysis, you need at least 1.8 volt, right? More or less 1.8 volt, but here maybe within 0.25 volt you can get hydrogen production. And you have to also remember that at the same time you are treating your waste and wastewater, right? So we are actually getting dual benefit. So we are treating our wastewater, extracting some energy or electron, and at the same time you are providing some additional electron to get biohydrogen. But why we need to apply voltage there? Okay, so look into, if we use simple acetate, okay, simple acetate as electron donor in our process, what is the redox potential? Minus 0.2 volt, sorry, minus 0.28 volt, right? So this is theoretical redox potential if you have oxidation of acetate, right? So if we provide microbes with acetate, minus 0.28 volt. But now if I want oxygen reduction to water, what is the redox potential for this reaction? It's 0.82 volt. But if you want to produce hydrogen, okay, if you want to produce hydrogen from proton, this redox potential is minus 0.41 volt. Okay, so now delta G we can calculate from number of electron transferred. Okay, and Faraday's constant, right, F, and we can have redox potential for acceptor and redox potential for donor. But even without calculation, what you see? So electron acceptor minus donor. So if I simply write, so delta G minus NF E acceptor minus E donor. So minus NF. Okay, let's talk about MFC. MFC, what is acceptor? 0.82, right? 0.82. And what is donor? Minus 0.28. Here, this is acceptor. Okay, so now this is going to be positive or negative, this delta G value? This is going to be negative, right? But how about for hydrogen? For hydrogen it's going to be positive, right? So that means for hydrogen it's not thermodynamically feasible because its delta G going to be positive, right? So what we need to do, we need to, and how much the difference? So minus 0.28, okay. So this is going to be, let me write it, let's do it for hydrogen because I want to show you how we calculate how much energy that we need. So here acceptor is minus 0.41. What is minus 0.41? And this is 0.28. So what is that? So minus, so that means minus 0.41 plus 0.28. So what would be that value?
Minus 30.
Right?
Minus 30.
Minus, yeah, minus 0.013 volt. So that means that's the energy difference. So that energy has to come from somewhere. Then you can have that reaction happen. So if you like provide 0.13 volt from outside, it's gonna reach an equilibrium, right? Delta Z is gonna be 0, right? But you need more than that. So minimum you have to provide some voltage more than at least 0.13 to actually kind of start the reaction to happen. Okay, so this is actually the thermodynamic significance of yes, you can decide, okay, I'm going to put this electron acceptor there and I want this product, but you have to look into whether you need energy to, you need to provide energy from outside or not. Okay, so here we are seeing that we need minimum of 0.13 volt, but if you provide 0.13 volt, do you think you are going to see hydrogen production? No. Why? Because there are going to be some other energy losses in the system. So there are going to be other resistance in the system that going to create some barrier for electron flow, right? So there are going to be more energy losses, and those energy losses we call them ohmic or internal energy losses. Okay, so they can be contributed by different component in your system. So for example, we have membrane, right, between anode and cathode, and anode and cathode, they are far from each other. That distance can also contribute to some energy loss. At the same time, like solution, you have electrolyte, right? That solution also going to actually like create some barrier for charge transport, right? So those energy losses caused by those factors are called ohmic losses. So that means if we provide theoretical energy that is required for reaction to happen, okay, that's not going to be enough. We have to provide enough energy to also make sure that that energy input can also compensate for these kind of energy losses in the system. Okay. So any questions so far? Okay. So I think yesterday I was talking about like we can combine this process with also like anaerobic digestion process. So for example, like anaerobic digester, we can introduce electrode and we can apply some voltage, okay, to generate hydrogen, okay, and we can have you know, like a better process. So how it can be better? So you can see here in anode we have electroactive bacteria, so they can actually oxidize you know, like a compound like acetate, okay, and then on the cathode surface some methanogen, they can either use hydrogen or some methanogen, yeah, they are hydrogen trophic, but some methanogen, they instead of using acetate or hydrogen, they can directly take electron, okay. Some methanogen, they can actually directly take electron, and it has been proved over last 10 or 15 years. So now yesterday we learned that actually there are two types of methanogen, right? So what are those two types? Acetoclastic and hydrogen trophic, okay. So there is third type of methanogen. Now we call them electrotroph. Why electrotroph? It came from, you know, heterotroph. What is heterotroph? They use organic carbon, and electrotroph means they directly use electron. So some methanogen, instead of hydrogen or acetate, they can directly utilize electron, okay. And you can see here, you can have this kind of reaction. So hydrogen can be produced from reduction of proton, and then that hydrogen can be used by hydrogen trophic methanogen. On the other hand, some methanogen, they can directly utilize electron from the electrode surface and produce methane, okay, and that has all.
I have a doubt. So yesterday you told us methanogen is not a good term, right?
Methanogen, no.
Because it's archaea.
Yeah, methanogenic archaea. I said we cannot call them bacteria. Okay, although they have similar feature like bacteria, we call them methanogen or archaea. Okay.
Methanogen is okay.
Yeah, methanogen is okay, but you cannot say methanogenic bacteria.
They are the type of bacteria, but
They are not type of bacteria.
they are actually not the bacteria. They are type of bacteria of
I guess.
No, they are type of microorganism, but they are not type of bacteria.
I
Okay.
guess it is just like bacteriophage.
No, yes, like as I was saying, like 30 or 40 years ago, researchers used to call them bacteria because they were almost similar, you know, like metabolic process, similar size and shape like bacteria. But later on, they found actually they are different. They are not bacteria.
But the archaea we only can find in extreme conditions.
Extreme, they can survive in extreme condition, yes, but they are not bacteria. Okay. So yesterday what I was telling that we cannot call them bacteria. We can call them methanogenic archaea or methanogen. That's fine. Okay. But here I am saying, because the thing is that every, you know, like in every 10 or 15 years, researchers, they find like new feature of microbes. They try to give them new names, right? So for example, electrotroph, it's kind of like, you know, it became very popular term in over, I would say, last 10 years. Okay. So if some microbes, they can take electron directly, we can call them electrotroph. Okay. But you know, nonetheless, so what you have in if you have MAC-AD, so in the conventional AD, what happened? We learned what happened. We have hydrolysis, right? You have fermentation, and then acetate is consumed by acetoclastic methanogen, and hydrogen and carbon dioxide used by hydrogen trophic methanogen, and you have methane production in your digester. But when you have MAC-AD, what would happen? Those acetate gonna be mainly utilized by electroactive bacteria, and that is kind of like desired. Why? Because we know that the growth rate of acetoclastic methanogen is very low. Even compared to hydrogen trophic methanogen, their growth rate is very low. Okay. But if you compare EAB and acetoclastic methanogen, you can see, so for example, Ks value for EAB is 20 to 190 milligram per liter BOD. But for Ks value for acetoclastic methanogen, if you remember from our day 2 slide, from 180 to 450. So which one is kinetically efficient? Low Ks value, right? So if something has low Ks value, this is kinetically more efficient. So that means if someone, so it's like, let's say you can run faster, right? So you can beat me, you can outcompete me. So the same thing happen when you have EAB kind of growing in your electrode surface, they can outcompete acetoclastic methanogen for substrate.
So EAB is nothing but ETB, electrotrophic bacteria, right? EAB.
Electroactive bacteria, EAB.
Kind of whatever we have studied.
Yeah, I know that it's confusing. Like
It's
so,
not confusing, it's
if
easy for me.
so, EAB typically we use if something can transfer electron, okay, like extracellular using EET mechanism, we call them EEB. If something can take electron, okay, by extracellular electron transfer, then we can call them electrotroph, okay. So yeah, many terms in this area.
EAB a subclass of ETB.
No, that is different because EAB is on anode side and ETB on cathode side, right?
No.
ETB on cathode side, yes. ETB, I never use the term ETB. So electrotroph means they are taking electron, and electroactive means they are transferring electron. Yeah. So actually EAB you can actually have like perfect actually mono pattern. We are going to actually discuss about that in later slide. But as you can see here, so what is for aerobic heterotroph? In day 2 actually we discussed that, right? So aerobic heterotroph in activated sludge process, their Ks value is from 12 to 60 milligram per liter BOD, right? So still those aerobic activated aerobic activated sludge process microbes, they might be kinetically more efficient, but compared to conventional anaerobic microorganisms like methanogen, that EAB, they are kinetically more efficient, okay. So still that's why researchers, they are thinking that these kind of microbes, okay, we can have anaerobic process for wastewater treatment, okay, and we can have possibly achieve low BOD concentration in the effluent, which may not be possible with regular anaerobic process. So another application could be microbial electrosynthesis, because these days there is lot of interest on like converting carbon dioxide, carbon capture, right? So converting carbon dioxide to some value added product, okay. So and we can also do that using this kind of process, but here your anode gonna be abiotic. So you can have anode, we can have water electrolysis reaction that can release electron, and electron can be transferred to the cathode surface, okay, where you have methanogen, like same as MAC-ID system, but here it's kind of separated by membrane, okay. And if we provide biogas, those microbes, they can hydrogen trophic methanogen, they also need carbon dioxide, okay. Electrotroph means they can take electron, but they also need a carbon source, right, for their growth. So they can use CO2 as their carbon source, and you can have almost like you can get rid of all the carbon dioxide, and you can get close to renewable natural gas. And some of you attended yesterday's research seminar, and actually I was showing even from our experimental data, actually we can completely get rid of carbon dioxide from biogas using this kind of process. So another application could be we can actually produce some value added chemical on the cathode, okay, and we can use that chemical for different application in wastewater treatment. So for example, one compound is hydrogen peroxide, which is a very strong oxidant, right, that can also be actually used for disinfection, okay. So how we can actually produce hydrogen peroxide? So you can see here typical MFC, what we have oxygen and 4 electron and 4 proton, they are combined together and produce water, right, and this is 4 electron reaction. But if you can also have, if you can manipulate or you can have a better catalyst, you can also have 2 electron reaction. So for example, 1 mole of oxygen, 2 mole of electron and 2 mole of proton can combine together and produce hydrogen peroxide, okay, hydrogen peroxide. So and then that hydrogen peroxide can be utilized in situ for different water or wastewater application. But this topic is, there are a lot of challenges, and why it's challenging, because already the proof of concept is done. Okay, there have been actually a lot of paper on this topic, but the major challenge was actually most of the time initially you are gonna see lot of peroxide production, but over the time peroxide gonna be decomposed to water again. Okay, so when you have lot of electron over time, so you are gonna see they are gonna be, so first you are gonna have 2 electron reaction. Okay, but when you are gonna have more electron, you are gonna actually, that peroxide gonna be decomposed to water again. Okay, so then you may not, so the one technique could be, as soon as it's produced, you kind of utilize them in situ. Okay, so you don't wait. If you wait, okay, you know what, you are going to extract the chemical, leave it, it's going to be decomposed to water. So that's why researchers thought, okay, still this could be a feasible approach, but you have to use that hydrogen peroxide in situ. Okay, so so that means your cathode chamber, okay, anode chamber can be a regular anode chamber where you are going to have EAB, they are going to use organic compound and you know, like transfer electron. But cathode, we can actually have we can actually feed our wastewater, okay, with different types of contaminants. So for example, peroxide, if we add iron salt, that is actually a strong oxidant, we call Fenton reagent. Anyone familiar with Fenton reagent, right? Yeah. So peroxide, it's a strong oxidant, but if you add iron, okay, we call it Fenton reagent, which is even better oxidant. Okay, so if we have, and that can actually, oxidant can generate hydroxyl radical. Okay, so and you can actually use that thing for advanced oxidation process, and I'm pretty sure some of you might be familiar with advanced oxidation process, right? So what we can do wastewater treatment. If we have very recalcitrant compounds, something cannot be degraded biologically, we can actually use Fenton reagent or hydroxyl radical to degrade them. So for wastewater treatment plant, the final step, what is the final step?
Chlorination.
Disinfection, right? So we can actually have even the final effluent, we can actually pump to the cathode chamber and we can have disinfection there, okay, in situ disinfection. Sometimes that hydrogen peroxide, some researcher also demonstrated that, okay, you know what, like anaerobic digestion process, sometimes hydrolysis can be rate limiting. Okay, so we can use this kind of strong oxidant to hydrolyze the organic, particulate organic. Okay, so even we can use this method to hydrolysis of like sludge or any other feedstock that we use for anaerobic digestion process. Okay,
So kind of pre-treatment.
kind of pre-treatment, yes. Any questions so far? Okay, so let's move on. Another thing we can do is desalination. So every year we see new application, okay, in this topic. But why I'm laughing, I'm gonna tell you later. But so how we can actually achieve desalination? Okay, so desalination we can have instead of 2 chamber. So far what we have seen, like 2 chamber, right, anode and cathode. But for desalination, between anode and cathode chamber we can introduce a third chamber. Okay, and why third chamber? Actually third chamber can be used for desalination. Okay, so how we can achieve desalination? So we can put, so typically anode and cathode chamber, they are separated by membrane, right? But what we have, so let's say, you know, like seawater, what we have? We have sodium, we have chloride, right?
And bromide.
And bromide. So basically some cations, some anions, right? So what if we put an anion exchange membrane in one side of this chamber and another side we put cation exchange membrane? Okay, so what would happen when you are going to introduce saline water? That anion exchange membrane going to promote transport of anion from this chamber. And why? Because of concentration gradient. You don't have enough chloride in there, right? So because of concentration gradient, that chlorine gonna transfer from the middle chamber to the anode chamber. On the cathode side, you have cation exchange membrane. So sodium can actually transfer from this chamber to the cathode chamber to because of concentration gradient. Okay, so that means you can get rid of, if you kind of recirculate continuously over time, you are going to see all chloride and your sodium kind of like transferred, they kind of disappear from the water, and now you have actually desalinated water.
So if you remove microbe from this system, it will be nothing but electrodialysis. It is a same
Yes.
system.
Yeah, so lot of feature actually adapted from other electrochemical processes. Okay, so saline water, and similar concept, actually a researcher, they demonstrated that we can actually get water softening. So for example, like in some cities, water can be really hard. Okay, water can be really hard, so you are going to see your plumbing system can be damaged over time, and sometimes when you have hard water, you don't feel good after taking shower, right? Your skin is very rough, feel very rough, right? So that's why water softening is a very popular process. But most of the time, water softening process, it's kind of like done by using resin, right? Using resin, and you use salt water to actually regenerate the resin. So which is a common process. But actually researchers demonstrated that if we actually provide hard water in the middle chamber, we can get rid of the ions like that contribute to the water hardness, like calcium, magnesium and other minerals, and we can get soft water from the process. Okay. So another thing we can achieve is nutrient removal and recovery. Okay. So how we can do that? So let's say we can have source separated urine. Okay. So we can have source separated urine, and in our urine what we have? Nutrient. We have nitrogen, we have phosphorus, right? So we have nitrogen more or phosphorus more? Nitrogen more, right? So what we can do, we can feed urine in the anode chamber of the MAC reactor, and we can have a cation exchange membrane. So if we have cation exchange membrane, ammonium, it's cation, right? So because of concentration gradient, some cation like ammonium gonna transfer from anode to the cathode chamber. Okay, so then you can actually collect those ammonium from your cathode chamber, and that can be used as a liquid fertilizer. But now phosphate, okay, this is anion, right? So phosphate cannot actually move from anode to cathode. So in the effluent you are going to have phosphate, okay, and you are going to have some remaining ammonia as well. Okay, so all ammonia are not going to be transferred, there are going to be some ammonia left. So if you actually collect the effluent and if you add some magnesium salt, you can actually get struvite, and I think some of you might be familiar with the term struvite. So struvite is actually magnesium ammonium phosphate, which is actually considered as a slow release fertilizer. Okay, so using this process we can also produce fertilizer. So we talked about denitrification, right? So what is denitrification? Nitrate to nitrite, right? So we can actually also achieve denitrification here. So how we can do that? Because some denitrifying bacteria, they can use hydrogen as electron donor, because denitrification process, you remember that we need to provide electron donor from outside, right? So because a nitrate is kind of like act as electron acceptor, okay, in anoxic process. So we can produce hydrogen, and if we put some denitrifying bacteria and if we provide nitrate, we can actually convert nitrate to nitrogen gas using denitrifying bacteria.
So could you explain this again?
Here?
Yeah.
So here it's going to be MAC. So you are going to provide power supply, okay, and you are going to produce hydrogen, and you are going to add some denitrifying bacteria, okay, they can form biofilm there. So you are going to have in situ hydrogen production, okay, and in cathode feed it's going to be maybe some water, wastewater coming from your aerobic tank, okay, or aeration tank, where actually you already converted ammonia to nitrate. Okay. So then you have denitrifying bacteria, you have nitrate from wastewater, you have hydrogen from the cell mag, okay, which is electron donor, and you have electron acceptor nitrate, and you can have denitrification. So basically, if I try to explain it more, so let's say you have electrode, okay, you have denitrifying bacteria. So from the reaction, electron is coming to the cathode, right? And you have proton, and you are actually converting that to hydrogen gas, which is an electron donor, hydrogen, right? And when you are actually feeding, let's say you have an aeration tank, you provide oxygen, you converted already all ammonium to nitrate, right? So if you pass this, let's say you have a cathode chamber, okay, you are actually feeding, you are recirculating between two tank, okay. So you are basically feeding nitrate there. So nitrate is electron acceptor there, and you have electron donor, and you have bacteria. So you have everything to get denitrification, right? So and then actually effluent can go back to your aeration tank for next process.
And in that effluent, what is there, which is going back to aeration tank?
Oh, it's then there is no nitrogen there. You completely remove them, right?
So clean one.
Yeah, clean water. Yeah.
And what happens?
It could be actually also used as a polishing step, right? Not necessarily you have to just. I was just showing an example, but it can be a polishing step.
And what happened to hydrogen here?
Oh, hydrogen is consumed in the process as electron donor. Yeah. Actually, there are some research on hydrogen, using hydrogen as electron donor. Actually, I think one group, even the lab I did PhD, actually they are also working on hydrogen as electron donor, but there are few groups actually working on that. So another application could be biosensor. Okay. So what is biosensor? Because those microbes, okay, so they kind of follow monod pattern. Okay. So if you increase the substrate concentration, the electrical response gonna increase. Okay. So if you actually increase the concentration, so you can actually get a perfect calibration curve. Okay. So let's say so what is monod pattern? So monod pattern means if, so what we learned, you have substrate concentration, let's say COD, right? And what is monod pattern? With increase in substrate concentration, growth gonna increase and reach maximum, right? So now this is growth, but here instead of growth we can consider electrical current, because those microbes, they provide like responses electrical current. Okay, so we can delete that, okay, and we can put current density. So if your COD or any contaminant level in within this range, okay, you can actually have a perfect calibration curve. Okay, so and you can actually use that concept as a biosensor. Okay, sometimes we kind of use as a biosensor for readily oxidizable compound. Okay, but sometimes you might have toxic compound. So if you have a clean baseline and let's say like suddenly your current decreased instead of increasing, so you can tell, okay, you have some toxic material in your wastewater. So this concept can be used for like to monitoring whether there is any toxic like compound is coming with your wastewater or not. So
I have a question here.
yeah.
Suppose in this kind of system, because bacteria is there, so they are also using energy to grow and maintain their cells. So by increasing the number of cells, don't you think the baseline will be changed? Or electrical response?
Electrical response में
So,
so to answer your question, so first you have to actually reach a baseline. Okay, so if you have a continuous flow, so baseline means your wastewater, okay, organic content is same, right? They are getting same feed. You have biofilm grow and some there are going to be some decay. So it's going to be more like stationary phase, right? But now if you have something changed, okay, you might see like current going to increase, or if you have toxic compound, current might decrease, okay,
So we have
and you may reach a new baseline.
So we have to look for the decrement.
Yeah.
Right here.
It depends.
Because our job is to detect here toxic compound.
If toxic compound, yes, if you are targeting any toxic compound, current gonna decrease. But if you are targeting any, let's say you wanna see whether BOD is increasing or not, whether, so let's say I put this kind of system in the effluent line where actually I am discharging my treated wastewater in river, right? Now if you see one day current is 5 milliamp, but next day you see your current is 10 milliamp. So you might think, okay, you know what, your treatment process may be not actually working properly, right?
Because
So
more COD.
more BOD is actually going there, right? So we can actually apply this concept, and actually some commercial product using this concept has already been actually available in market. Actually, even I met actually one company few months ago. They are actually trying to actually we are trying to collaborate and see, you know, if we can actually kind of make some bio. So from my research group, actually we try to develop a biosensor, okay, for oils and process water. So we are trying to see if we can actually apply this concept, because most of the time oils and mining site, they are in remote site, right? So there you may not have like, you know, large analytical equipment where you can take sample and do it on site. But if we can actually develop something that we can actually work in, you know, like apply in the field, maybe they don't need to actually take sample and send it to those sample to some analytical lab. So based on the electrical response, they can kind of tell, okay, whether, you know, there is some seepage of any contaminant in groundwater or something happen in the process, okay.
I guess we can connect to the IoT and we can also get the data online.
Yeah, data online as well, yeah. So yeah, that part, so that's kind of what we are trying to do. So some commercial company, they are expert on this kind of, you know, like, so we can do bench scale research and kind of, you know, develop the technique, but we don't, you know, kind of develop the product, final product, right? So
Scalability.
yeah, scalability can, yeah, sometimes we need to collaborate. So this is, before I jump into like process parameter and kinetics, any question? Yes.
Sir, what will be the maintenance of the bioprocess? The number of microbes can be increased or decreased in such conditions.
If you have like you know, like if you have, let's say, maintain them with same concentration of electron donor, okay, it should actually like it should not change, because they are going to be, it's going to be more like stationary phase. If you see microbial growth curve, it's going to be more like stationary phase. So growth going to be same, growth and decay going to be kind of same, so it should be stable, okay. But why I was laughing, okay, so you can see there are a lot of you know, promising result, okay. There are a lot of different things that you can achieve with this you know, area, but still there are a lot of challenges, okay. And one of the major challenges is how you scale up this kind of system. So when you you know, do some experiment in the lab, you work with small you know, like a small reactor, right? Small reactor, that means your ohmic loss is small. But when you make a large reactor, okay, you have a significant energy losses in the system. So now the question is how we can scale up, because I can do experiment in, let's say, 500 ml reactor, but for wastewater I need few 100 meter cube, right? So how I achieve that? So that's kind of like the main challenging part here, and still I would say we are kind of in learning phase, okay, fundamental learning phase. What happened when that topic became very popular, okay, in early 2000, okay, researcher thought, okay, you know, when there is some hot topic, everyone want to do the same thing. Okay, so what they do, okay, wastewater A, they try to see, okay, it can produce current or not. Wastewater B, you try that as a, so more like focusing on treatability study, and those treatability study actually doesn't help that much to, so I must say, okay, you know, I'm not criticizing any researcher, but I must say this is not a good approach if we want to scale up something. So we should focus on fundamental and try to see what are the challenges and then overcome one by one. Yes, product like biosensor, because biosensor can be a small reactor, right? What I did in the lab, the same thing can be in the field, same size, right? So it's not that challenging, and that's why the idea of biosensor has already been commercialized. Okay, but if you are thinking about wastewater treatment for a community or something, then it could be challenging. We have to think about, okay, you know, how we can scale up this kind of idea. Okay. Any other question? All right, so let's move on. You want to take 5 minutes break? Okay, let's take 5 minutes break and then we'll continue. This is, I have another 15 or 16 slides. Okay, so let's take 5 minutes break. Start again. So let's continue. So far what we learned about some fundamentals, how microbial electrochemical technologies works. We learned about EET, how actually you can have extracellular electron transport, right? And then we learned about different application, okay? So how we can actually manipulate different types of electron acceptor, and we can get different types of validated product or other application like biosensor. So now we are going to talk about one of the important aspect, okay? So which is basically we are going to learn about significance of different process parameters in for microbial electrochemical systems, and we are also going to actually talk about kinetics, okay? So we learned about kinetics for activated sludge process, and we are going to see how kinetics can also be applied for this kind of process. So substrate affinity. So those electroactive bacteria, okay, they can directly oxidize some organic contaminants like acetate, okay. But if you have fermentable substrate, okay, so let's say you have some complex substrate like glucose or protein or like some other compound, okay, they need some sort of partnership with some other microorganisms like some fermentative bacteria. So what happen if you have some complex substrate like glucose, it's not going to be directly oxidized by EAB, okay. So you need some fermentative bacteria, okay, they are going to ferment, produce some organic acid, and then that organic acid can be utilized by EAB. So we call this mechanism syntrophic interaction with non-EAB. So some bacteria, they are not electroactive bacteria, they degrade and their metabolite can be used by EAB. So but most of the studies in this area, okay, have been done with acetate as a substrate, okay. And why is that? Because we know that acetate can be directly utilized by microbes. So for some fundamental study, okay, acetate has been widely used as a model compound, okay. But for real life, you have to make sure that you have a balanced microbiome in your biofilm. You have both non-EAB like fermentative bacteria, and you have electroactive bacteria. So especially when you want to design something for real wastewater or complex wastewater, okay. Another major concern in this kind of process is proton accumulation in the biofilm, because if you remember your anodic reaction, if you have acetate, okay, that's going to be oxidized to carbon dioxide. The process also going to produce proton. You can see 1 mole of acetate going to give you 7 mole of proton and 8 mole of electron. So that means you expect that, okay, when electron is moving from anode to cathode to maintain charge neutrality, your proton is also going to move from anode to cathode, right? But in reality that thing may not happen, okay. So that thing may not happen because that proton also concentration gradient is also important. Some other cations, okay, for some other cations concentration might be high, and instead of proton those other cation may actually move from anode to cathode to maintain charge neutrality. So not necessarily it has to be proton. Yes, we expect that ideal case is going to be proton, but in reality that may not be the case. So what would happen if proton is concentration kind of proton accumulate in the biofilm? It's gonna increase or decrease your pH? Decrease, right? If you have higher proton concentration, then your pH gonna decrease. But those microbes, they prefer neutral pH, right? But now let's say some microbes, they are going to be in inner layer of biofilm, some microbes can be in outer layer. For outer layer, maybe proton can easily migrate from the biofilm, okay, or maybe diffuse out of the biofilm and go to the cathode. But some microbes that actually they are doing this reaction and producing proton, maybe inner biofilm, that proton may accumulate, okay. Then those proton pH gonna drop and those microbes may be inhibited. So you might have a different zone, okay? You might have a different zone. So inner layer could be like inhibited or inactive zone, like dead microbes, and outer layer could be like more active bacteria. So that thing could happen as well. So that's why, like, you know, more thick biofilm, sometimes what we think, like, you know, if we have more active biomass, we have better process, right? But that may not be the case. You might have an inactive zone and active zone. Although actually when we do lab scale studies, we add buffer with our, you know, say electron donor. So we don't see pH drop. But if you don't have enough buffer in your wastewater, then that can be a problem, okay? So like during my PhD, actually I did some experiment. So, you know, like decreasing buffer content, and what I did, we had some sort of like microsensor. So we could see a different depth of biofilm, what is the pH, okay? So and I could see actually pH dropped near the actually electrode surface. And then we also did some, you know, like confocal microscopy with, you know, staining the biofilm with, you know, those live and dead cell that you could see. And I could easily see, like, you know, there are a lot of red cells. So that means dead cells inside of the biofilm, and outer layer is more like green cells, okay? So pH accumulation, proton accumulation or pH drop could be a major challenge for this kind of process. The same thing could happen for substrate, okay? So if you have a very thick biofilm, okay, outer, you know, like outer layer of biofilm, those microbes, they can easily get substrate. But maybe inner layer, there might be some substrate limitation. And you can have same, you know, problem. Like you can have some substrate limited zone, and you have inactive zone and active zone. So that's why actually the biofilm thickness or maintaining certain biofilm thickness that can actually avoid proton accumulation or substrate limitation, it's very important, okay? So thick biofilm doesn't mean that, okay, you know, you have more active biomass and you can have better performance. So sometimes actually, you know, like some researcher, they applied model like, you know, finite element method to actually, you know, kind of model this kind of substrate gradient inside the biofilm, okay? So those are actually kind of like, you know, combining Monod equation and Fick's law for diffusion, okay? So you can combine different biological model and diffusion model combined together and see whether you are going to have some proton accumulation inside the biofilm or substrate limitation, okay? Another way is you can use microsensor, but the microsensor typically they are quite expensive, okay? So now we are going to have a very important topic. So on day 2 we learned about kinetics, okay? So we learned about kinetics, and what we did, we kind of used Monod equation. So what Monod equation is telling us? Can anyone volunteer? What is Monod equation telling us?
Kinetics of microbes.
Kinetics of microbes, yes. So this is actually Monod equation. If you remember mu max, anyone remember the equation? Ks plus S, right? So this equation is saying, okay, you know, like when substrate concentration is limiting, okay, so with increase in substrate concentration, growth gonna increase. So graph gonna look like this, mu, and at a certain substrate concentration, growth gonna reach maximum, right? That's Monod equation. So now researcher, they try to see whether, okay, we can apply that Monod equation, okay, and we can actually estimate. So one of the thing, output from the system is current density, right? So we kind of like, you know, microbes, they oxidize organic compound and then they actually produce current, right? Current density. So they are trying to see if substrate is only the rate limiting part, if we can actually describe this system using Monod equation or not, okay? And actually it was, well, I'm sharing it here, that means actually we can do that, right? So, so that's why I was telling, if you learn the kinetics or how to establish, you know, kind of model equation for one biological system, you can apply, then you can actually learn other biological system very easily. And that's actually the beauty of environmental biotechnology. Okay. So let's see how we can actually apply Monod equation for EAB biofilms, electroactive bacterial biofilm. So current profile to substrate concentration follow typical Monod pattern, okay, which is typical Monod pattern, when substrate concentration is the rate limiting factor for current generation. So here we are seeing we are going to have substrate is the rate limiting, okay, but here growth, actually growth part, we are saying instead of growth we can consider current density is the output, okay. So we use J for current density. Okay. So now, so the substrate utilization kinetics for microbial electrochemical system has been successfully modeled with Monod equation. And Monod equation, what we learned, substrate utilization rate equal to substrate, okay, minus Ts by dt equal to what, substrate concentration, right? Okay, now we can put a proportionality constant, okay, mu s, right? So this is kind of like Monod equation, substrate utilization kinetics, right? So now you can see here, sorry, it depends on the not S, actually it's a biomass concentration, yeah, sorry. So mu max. So do you agree with that? It's good? Did you have a chance to go through the material that we shared? So substrate utilization rate, okay, it depends on the, it's proportional to the concentration of biomass, right? So yeah, dx by, it's changes substrate utilization rate, ds by dt, but it's proportional to x, right? And we can put a proportionality constant which is mu, right? So this is a specific substrate utilization rate, but here instead of mu we are going to use q. So q is the maximum specific rate of substrate utilization. So what I am saying here, maybe I clean the board and start writing from scratch, I think that would be better. No, we cannot directly replace, we have to do some concept, because yeah, so one is current, one is COD, right? So we cannot directly switch. So RSU equal to so basically ds by dt, okay. I can put either minus here or minus here, okay, it doesn't matter, it's decreasing over time, which is proportional to the, proportional to the concentration of biomass, right? Concentration of biomass. But now what I am saying, so mu, so typically what we write minus RSU equal to mu x, right? But mu x, but here we are saying mu is for like regular heterotrophic microbe, okay, but for those electroactive microbe we are going to use a term called q, okay. So just you can put mu, but they decided, okay, you know what, there's a different types of microbe, so we use a different term q, okay. And so now we are saying that x, this substrate utilization rate going to depend on, okay, So for activated sludge process we consider the X, right? But here we are saying this is going to be XA, active biomass. Okay, so it's going to depend on active biomass. So it is almost the same thing what we did for activated sludge process, but just different, like I would say different symbol that we are using. So XA, active biomass, and Q is substrate utilization rate, right? So now we know from Monod equation Q, Monod equation we know mu equal to mu max S Ks plus S, right? So here it's going to be Q max S Ks plus S, right? Just the only thing I am replacing mu with Q. So now what I can write, Q max XA S Ks plus S. Can we write this? Make sense? So exactly Monod equation, just I replaced mu with Q, okay, and then I put it there. So now you see, and instead of X I put just XA, active biomass, okay. I could put X honestly speaking, but this is the one that model who developed the model, he put XA. So just to respect him, I am also putting XA. Other than that, it's nothing else. So it is the same thing that what we did. So now, so you can see we have almost a similar thing here. Okay. So this came from the Monod equation and substrate utilization rate. Okay. So now let's see next step. So now we are saying that XA, active biomass concentration for a biofilm, okay, can be calculated from EAB concentration in biofilm, which is, so now we are saying that okay, this active biomass XA, but we can actually estimate that from concentration of EAB biomass, which is XF, okay, XF and biofilm thickness. So if you multiply by the concentration of electroactive bacteria, okay, SBSS, by the thickness of biofilm, because it's a biofilm system, it's not a suspended growth process, right? So that's why we want to multiply the concentration by the thickness of the biofilm. So what we are saying, XA, I can replace that with XF and LF, which is thickness of biofilm, and this is concentration of EAB biomass, okay. Make sense? So now what I can do, I can replace that part with XF and LF. So I did that here, XF, LF. Make sense? We're good? Okay. So now what we are saying, now substrate, when substrate is rate limiting, then current density will follow Monod pattern. So what we are saying here, this is a special system. Instead of growth, okay, substrate concentration, we can have actually, we can replace that with current density, with substrate utilization rate. So what about, in that case, what about the substrate utilization rate or substrate flux, okay, it's gonna be actually current density in the system. So now so substrate utilization rate, this term RS, I'm going to replace that with JS. So kind of, now we are saying it's a flux of electron donor. Okay. So now RSU term, I'm going to replace with J sub for substrate. So substrate flux within a, for certain area. So in the biofilm, okay. So this is for biofilm. So substrate flux within the biofilm gonna be same as the substrate utilization rate, okay. So J sub gonna be this. Okay, now we are saying that, okay, you know what, like substrate flux, okay, gonna be equivalent to current density. But one thing is, so current is Coulomb, right? Coulomb per second. If you say ampere, so it's Coulomb per second. So it's electron transfer rate. But our substrate in biological process, it's COD, right? So one thing is COD and one thing is current. So that means I cannot just directly consider, okay, this is my current, right? I have to introduce some term that can allow me to convert COD to current. And how I can do that? I can actually introduce another term. You can see here 0.14 Fe, right? So now let me clean the board. This is a key part here. Okay, now what I am saying that I can convert that. So that was COD flux, right? That is COD flux, but I want that to convert it to current. So how I can convert that into current? So I can introduce a term 0.14 Fe naught, okay, and then Q max XF LF S Ks plus S. So why 0.14 Fe? Okay, so 0.14 Fe, it came from two things. So when microbes, okay, so Fe, you remember that, you know, when we did half reaction including cell, we had what? Fe and Fs, right? So Fe plus Fs equal to 1, right? That's what we used. So a portion going to be utilized for cell synthesis, a portion going to be utilized for going to go to the donor, right? But here my product is current. So a portion of electron going to go for cell synthesis and a portion of electron going to go for current generation in the process. So we are introducing a term, okay, it's not going to be 100%, a fraction of electron or COD going to be my current, okay. So and it could be that Fe naught could be 0.9, that Fe naught could be 0.8, depending on, you know, like how much is going for current generation, okay. But why 0.14? That 0.14 is the conversion of COD to ampere, okay, and you can see here I put it there. So 1 mole of electron per 8 gram of COD. So COD, 8 gram of COD is equivalent to 1 mole electron. How? Do you know?
This one
Yeah, 1 mole electron.
from half cell reaction.
So from half reaction. So for example, oxygen reduction to water, right?
Yes.
How many proton I should put here? 2. And how many electron?
2 by 2, I guess, because when you balance oxygen, there H2O you have to multiply by 1 by 2. Multiply by 2.
2, right?
Then it should be 4.
It should be 4, okay.
And that one also, proton also 4.
Proton 4, okay, now it's balanced, right? So now oxygen, it's how many gram of COD? Hmm? 32, right? So 32 gram of COD. So 32 gram of COD is equivalent to 4 mole of electron. Can I write this?
Yes.
So now 1 mole of electron,
8.
8. So 1 mole electron means 8 gram of COD, right? So that's exactly what I wrote. 1 mole electron, 8 gram of COD. So now per mole electron, what is the charge? It comes from Faraday's constant, right? So Faraday's constant is saying like 1 mole of electron gonna give us 96,500 Coulomb, right? It's a value for F. So now what is ampere? Ampere is Coulomb per second, right? So day I can, in one day, I have how many second? I have 86,400 second, right? So now what I am getting, 0.14 coulomb per second. So coulomb per second is ampere, so I can actually replace coulomb per second with A. So 0.14 ampere is gonna be equal to 1 gram COD per day. So that's why I put 1.14 here. Okay. So now we are good to go. We converted everything. Our COD has been converted to current. Okay. So now that equation can be directly used. So what if we put kinetic parameter here like Qmax, XF, and biofilm thickness, if we put substrate concentration, if we put Ks, what we are going to get, it's m per meter square. Okay. But previously in this equation, it was substrate flux, it was more COD. You see here, gram COD per meter square per day. But here, it's ampere per meter square, right? So we already converted from current to, COD to current. Okay.
Current density.
Current density, yes. So now, what would be the value for Fe? How I can get that?
1 minus Fs.
1 minus Fs, but how you are going to measure Fs?
The previous concept.
So Fs sometimes it is difficult to measure. Okay. So what we do, we have a term called, so electron donor is going for end product. Okay, and another portion is going for cell, right? So here my end product is current. Okay. So if I know the COD removal efficiency in my system, how many gram of COD removed, I can convert that into coulomb, right? I already, we know how to convert COD to coulomb, but now I'm going to have some experimental data where I'm going to have current. I can also, current means it's ampere, right? Ampere is coulomb per second. So if I know the current, I can also convert that current to coulomb, right? So I have some, how many coulomb I should get from my COD removal and how much coulomb actually I'm getting experimentally. From this, I can calculate something called coulombic efficiency. So coulombic efficiency is the ratio of total coulomb actually transferred to the anode from the substrate to maximum possible coulomb if all substrate removal, actually all substrate converted to current. Okay. So Q coulomb and that is coming from current, and this Q substrate is basically how much COD removed in the process. Okay. So typically this is going to be less than 100%, right? Because anaerobic microorganisms or any kind of like aerobic process, you are going to have more sludge production, right? So only small portion, like 60%.
It's not like efficiency, it's more like yield, right?
It's more like you can say yield of current.
Yes.
Yeah, it's more like yield of current.
Because it would be efficiency, then we have.
But yield, I don't want to use the term yield because sometimes yield is used for like how much biomass produced, right? So yield is how much biomass produced per gram of COD removed, right? So we don't use the term yield. We use, so if you have, let's say, coulombic efficiency is 90%, my Fe, this one gonna be 0.9 and this is gonna be 0.1 because I cannot, it's not very easy to measure, right? If my coulombic efficiency is 80%, I'm gonna consider my Fe naught is 0.8. If it's 70%, I'm gonna put 0.7. Sometimes coulombic efficiency can be much less than, let's say you can see 50%, but we expect that, okay, anaerobic microbes, okay, it should not go, your cell synthesis should not be that high. So that, what are the other electrons seeing then? So sometimes what happen, some non-EAB, some microbes, some other microbes, they can actually take some of the electron if you have a mixed culture biofilm, okay. So that could happen, but anyway, that's CE calculation or coulombic efficiency calculation can allow us to estimate Fe naught. Sometimes, but do you think your coulombic efficiency can be more than 100%?
No.
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 Actually, sometimes you might see it's more than 100%.
But
Okay.
It will defy that equation.
Yeah. So actually, even in one of my experiment, okay, that was done in back in 2012, okay, we found that actually continuously, like our Coulombic efficiency is higher than 100%. Then we went back to the literature and we found that, okay, what happened? Those microbes, you remember phosphorus accumulating bacteria?
Yes.
In anaerobic condition, they take VFA and store them as biopolymer. Those microbes can also do that. So when you enrich, you actually run the system with very high acetate, okay, concentration, and sometimes they actually store the acetate, okay, as some intracellular biopolymer, okay. And what happened later on, they start oxidizing those biopolymer and transfer electron. So that's why, yes, you see, like, your substrate is providing some electron donor, but you are getting actually more current than you should get. And that additional current might come from actually those oxidation of biopolymer. And actually this is actually an confocal image of we could see like some biopolymer accumulated within the biofilm. Okay.
So did someone explore this storing of electrons inside the
There are few paper we reported, but that was not our like primary focus. My PhD research was more focusing on kinetics. So I did not further investigated this. We knew that, okay, that thing could happen. We reported that, but we did not dig much. Okay. But yes, you might see. So if you see sometimes your Coulombic, if you're obviously, if you kind of in future, if you plan to work on this area, and if you see Coulombic efficiency higher than 100%, I think that could be one thing could happen. Okay, not necessarily it's something wrong that you did. So let's go back to our topic. So now we have this equation. Okay, so now if substrate is not rate limiting, so substrate is not rate limiting means your substrate concentration is very high. Okay, it's much higher than your Ks value. What is Ks? Ks is when actually growth is, or here it's current, right? It's Jmax, because we are not using mu. So your Ks gonna be Jmax divided by 2, right? At concentration where Jmax divided by 2. So if you are running your system at very high substrate concentration, then actually your Ks very higher than substrate concentration higher than Ks, then actually that part we can consider, okay, Ks is kind of very negligible, okay, compared to value for S. So that part I can consider. This is gonna be, so if S, Ks plus S, if I consider this is kind of quite negligible and this is quite big, this is gonna be 1, right? And at that time we can get Jmax, maximum current. So maximum current means Jmax equal to 0.14 Fe naught Qmax XF LF, because that term gonna be 1. So now I can actually put it back here for Qmax. Okay. So sorry, Jmax. So you can see this part we can actually replace with Jmax, right? So what I get, J equal to Jmax S divided by Ks divide by Ks plus S. So which is almost same as your Monod equation, right? So if your system only the substrate is rate limiting, okay, and if you run your system at different substrate concentration, okay, your current gonna follow this pattern. Actually this graph is from one of my experiment that I did. Okay.
So how did you do simulation here?
Oh, then we actually feed the model and see whether it can actually follow the mono. So those are my dots are my experimental data. Yeah.
And yellow is simulation.
Yeah, and yellow is simulation, and you can see it's kind of following. Yeah, yeah. But later on, researcher actually extended this model. They try to add more component, because this is Monod is more microbial metabolism, but in here we also have lot of electrochemical component, right? So then this model was further extended, combining some researcher combined that with Nernst equation, okay, for like electrochemistry. So that was actually combined. Sometimes they call them it's Nernst-Monod equation, okay. Then also some researcher actually combined that with Ohm's law, okay. So Nernst-Monod Ohm equation, okay. Even some part of my PhD thesis I kind of introduced Ohm's law, and I was measuring biofilm conductivity and see if we can actually fit that model as well, okay. But in most cases, okay, this part microbial metabolism or intracellular kinetics is the rate limiting. So other part, even if you add, okay, it doesn't matter, okay. So Monod equation in most cases can like I would say represent your system, okay. And we are going to actually, I have an example on this model, okay. So we are going to learn how to estimate Coulombic efficiency. We are going to learn how we can estimate current density using this equation, okay. But before that I have 2 or 3 more slides, I think. So current status of microbial electrochemical technology, as I mentioned, it's still in lab scale, but some product like biosensors have been actually commercialized, okay. Another product has been commercialized in Europe. So there is something called constructed wetland, okay. So they try to combine microbial electrochemistry with constructed wetland, and they did some actually demo, I think in some country in Europe, okay, couple of years ago. And but still, if you are thinking about waste or wastewater treatment, it's kind of still like we need to do a lot of work to scale up the process. And another thing, we always say, okay, you know what, that process can help us to recover energy as electricity, okay. Still current density maximum that we get from 10 to 25 amps per meter square, which is still not practically feasible. And this 10 to 25 amps per meter square or current density we can get with acetate, right, from your fundamental study. But if you use real domestic wastewater, it's still less than 5 amps per meter square, okay. And another challenge is how to scale up the process, because when you have, as I was mentioning, you have large reactor, means you have more ohmic losses, right. So that could be a big challenge here. So some researcher, they propose that, okay, you know what, instead of making one big system, why not we make like multiple small system, okay, and kind of add them in series or or parallel connection, or that might be a solution, okay. Another like if you look into this equation, if you have more active biomass or more EAB, okay, we can actually increase J or current density. So we are thinking, okay, what if instead of one electrode we put multiple electrode, but both it's still actually facing a lot of challenges. For example, even during my PhD I did a study with 4 electrode, and what I found, actually 4 electrode you don't linearly increase the performance, and 4 electrode they have different microbial community, different kinetic feature, okay. So it's kind of like quite challenging to achieve, like so putting 4 electrode not gonna give you 4 times performance. So we still have to do a lot of things in terms of design and in terms of fundamental as well, okay. So for example, like we have to better learn how that EET thing works, okay. Still we need to know about microbial competition, okay, syntrophic interaction, because if you want to use complex wastewater, then syntrophic interaction between EAB and non-EAB or fermentative bacteria are going to be very critical. So now the question is how we come up with a balanced biofilm, like combining all those different types of microbes, right? How to actually establish this kind of biofilm. And rule of biofilm thickness. Now we don't know, okay, how I can control the biofilm thickness. What is my optimum biofilm thickness, right? So this thing actually can have a lot of impact in your process. And substrate and pH gradient as well, okay. So this is actually those 2 two component, they are kind of intertwined: thickness, substrate, pH. So we have to do a lot of work, okay, and that's what we have been doing, okay. I would say 15 or 20 years ago, when you know, like people's first test, not I think 20 or 25 years ago, when actually people started working on this, it was very hot topic. Everyone jumped, okay. They wanted to do something with this, but then later on, you know, when they kind of found, okay, there are a lot of bottlenecks, okay. Now community is getting smaller and smaller, okay. In Canada, I think only me and I think my another colleague, just 2 groups we are working on this right now, okay. But it used to be very large group actually, research community. So, so yes, there are a lot of, you know, you can see, you can have lot of different application. It's a very promising and emerging, but at the same time, okay, it comes with lot of challenges, and we have to overcome that if we want to actually push it to the market. So before I finish, I have 2 example, okay, calculation, because I love calculation, okay, in my class. So let's see how we can calculate Coulombic efficiency, and we are gonna, the kinetic equation that we developed, we are gonna learn how to apply that kinetic equation to estimate current density from a system, okay. So the first example, a lab scale MFC 28 ml is operated under fed batch mode 35 hours with acetic acid 1 gram per liter. Based on the following information, calculate Coulombic efficiencies, okay. Current density is 0.2 milliamp per centimeter square, anode surface area 7.1 centimeter square, acetate is completely utilized by EAB. So to estimate Coulombic efficiency, what I need? I need, okay, how much Coulomb I got from substrate, right, or electron donor, and how much Coulomb I am getting from as electricity, right. So the ratio of both can actually give me like Coulombic efficiency. So now, so Coulomb, Q Coulomb, which is basically coming from current density, and Q substrate is basically coming from your acetic acid. So now Q Coulomb, so current density is 0.2 milliamp per centimeter square. So now ampere, so Coulomb is basically ampere is Coulomb per second, right, Coulomb per second. So I have to convert that current into Coulomb. So how I can do that? So I have milliamp, 0.2 milliamp per centimeter square. So what I am going to do, I am going to convert that into amp. So I can divide that 0.2 by 1000 milliamp, and it's going to be amp. And I have the, so this is current density, right. So I have to multiply by the anode surface area, which is 7.1 centimeter square. So I multiply by the centimeter square, 7.1 centimeter square. So I get rid of centimeter square and centimeter square here, right. So I only have milliamp. Now I operated the system for 35 hours, okay, 35 hours, but current is Coulomb per second, right. So I have to convert that hour to second. So 3600 second equal to 1 hour, right. So now what I am getting, 178.92 Coulomb. I don't need, because I am getting rid of it, right. So what I am you got it? Yeah, I don't need to convert this, because I have centimeter square, and centimeter square I can just, you know, cross out. So now this is actually coming from current density. So how much Coulomb is coming from the substrate? So we have 1 gram per liter of acetate, and that is completely utilized, right. And we have 28 ml the volume, right. So volume multiplied by concentration is I can get mass of COD, right. But now this is 1 gram per liter of acetate. So I have to convert that concentration to what? COD. You remember in second day 2 we learned how to actually estimate COD equivalent for some compound? And what was your first practice problem? Did you have a chance to have a look? So first practice problem was estimate COD equivalent for acetate, right? So what was the COD equivalent for acetate? 1.07 gram of COD per gram acetic acid or acetate. If you go back to the day 2 lecture note and if you check your practice problem, it's there. We already did that. So COD equivalent 1 gram of acetate gonna give you 1.07 gram of COD. So that's why now I put 1.07 gram of COD and I have the volume of the reactor. So concentration multiplied by volume, so I can get the mass of my COD, right? So 0.028 liter, and now COD need to be converted to what? Coulomb. And we know that 8 gram COD, 1 mole electron is 8 gram COD, right? So now what I do, I convert the COD to mole of electron. So 1 mole of electron per 8 gram of COD. And now I know the charge of electron. So 1 mole of electron from Faraday's constant, it's 96500 coulomb per second, right? So sorry, per mole of electron. So now I am getting, this is the amount of coulomb I should get, okay, if I completely recover all the or completely utilized all the COD for current generation, right? So this is what I should theoretically get, but I am getting practically 178, right? So my Coulombic efficiency is 178.92 divided by that theoretical coulomb 361.39. So basically 50%. So that means 50% of the COD, okay, point for something else, cell synthesis, maybe some it utilized by other non-EAB microbes, okay, and only 50% we recovered as current in the system, okay. So that's kind of also allow us to estimate what is Fe naught, right? So the second problem, and that's the last slide for me. So calculate the maximum theoretical current density for a MAC fed with sufficient acetic acid. Again, acetic acid means 1.07 gram COD per gram of acetate, and Coulombic efficiency is 80%. So my Fe naught is 0.8, right? So in this equation my Fe naught is 0.8. And other kinetic parameter, I have Qmax 540 milligram acetate per gram VSS per hour. I have XF, which is 2.8 10 to the power 5 gram VSS per meter cube. LF, which is biofilm thickness, right? So I have value for Qmax, I have value for XF, I have value for LF, right? So first thing we have to do, you can see here Qmax, this is given as acetate, but I need to convert that into COD, right? So what I am going to do, 540, I am going to first multiply by 1.07 and convert that into gram per gram VSS, okay, because I think other thing are provided in gram, right? So it's better to actually convert that into gram. So 540 milligram acetate per gram hour, I am getting 0.58 gram COD per gram VSS per hour. Okay, so now, so this is my equation, okay, substrate is not rate limiting. So they are fed with sufficient acetic acid. So that means I am considering Jmax, right? So my substrate is not rate limiting anymore. So now 0.14 Fe naught is 0.8, right? Because Coulombic efficiency is 80%. So this is going to be 0.8. Qmax we already converted. XF is given 2.8 into 10 to the power 5 gram VSS per meter cube. LF I already have 5 micrometer. So 0.14 multiplied by 0.8, I have Qmax, I have value for XF, the biofilm thickness, I have to convert that into meter. It is provided in micrometer. So that's why I divided by 10 to the power 6, and now you need current. So that means it's gonna be per second, right? So sorry, it was in hours. So I can actually convert hour to day, right? So I can. So you have 2.18 amp per meter square. This is the current that you can get from your system. So that means if we know the kinetic parameter, if we know the Coulombic efficiency, we can estimate what is the current. Okay. So that's kind of a simple approach to model this kind of system. Okay. Any other question? Very good. There is some unit issue.
You can recheck later.
Okay, I can check later. Yeah.
In that hour day, because it should be opposite. Because Qmax we have per hour per number. So basically, you are trying to replace hour, cancel out hour somehow.
Oh, actually, why I put that? Because 0.14A is basically gram COD per day. That's why a hour was converted to day.
Okay, so here the unit is missing. 0.1.
Yeah, the unit I did not put. Yeah, yeah. Yeah, so basically, 0.14A, when we put that, this is for 1 gram COD per day, right? So that's why I converted hour to day.
This model is good, sir, but has so many limitations.
Yeah.
And has it been validated for any available data
Actually, yes.
to put
It
into practice?
you can. So I can tell you the graph that I put here. Actually, this is my experiment from my own experiment. Okay. So yeah. So that means you can say model actually works.
That is well and good, sir, but we have kept so many limitations here.
Yes. Yeah.
Hypothesis.
So it's a biological system. It's a very
Yes.
complex system, right? So.
With the electron results, this cannot be implemented in practice.
So what kind of limitation you think that we need to overcome?
So for example, Ks. S is far more than Ks. That is one limitation.
So how it can be a limitation? You can provide substrate more than
That is
Ks.
only this J is equal to J max SD by Ks. It follows monotonicities.
Mhm.
So like that, so many other thickness of film. Thickness of film is a variable in fact. That depends upon normal.
But you are actually using thickness in your model.
That is also a variable.
Yeah, that is also. So if you change your LF, obviously your number gonna change, right?
So like that, some hypothetical, reasonably acceptable assumptions are there. My idea is, if this model is holding good, it has to validate with the experimental results. Has
It already has been validated with experimental result. What I am saying, this is my experimental result. You can see I feed that model there.
That is what is my plan. My idea is, in practical situations, has it been implemented?
So that's what I was telling in my very last slide. Okay,
That is not a limitation, that is a condition in which it applies.
That's what
so if you see practical, if you say
it's an ideal condition where all the conditions are defined.
so if you say practical application, practical is more like scale up issue. It's not about like model. It has nothing to do with model.
So for practical application, he already mentioned about biosensors. So it's
And-
already, and he also mentioned about a plant in Europe where he can also do the wastewater treatment, actually in sync with the wetland, right?
Yeah, and
So
one
those are
thing
the
is
practical-
like you can go with instead of, so the major challenge is when you develop or fabricate big system, you have more energy loss, right? So now one thing could be you have multiple unit, but obviously multiple unit gonna cost more. So typically one big system costs less, right? Multiple small things actually going to cost you more. Now we don't know whether it's feasible or not. Another thing could be like we put more electrode, but we know that actually that doesn't work. It has been actually proved in even in bench scale it doesn't work. So how you expect that it's going to actually work in full scale, right? So it's not about a model problem. Actually Monod model has been applied. So why I introduced that in the class, I was telling you that actually like this kind of kinetics, if you learn for one system, you can actually apply for other system as well. Let's say not necessarily, you know, like what topic we covered in this course, okay? Your research topic is something different, but you can take this fundamental learning and you can apply it for something else. And as you can see, like, you know, this is an emerging topic, but we can still use that Monod equation and apply that to estimate current density, which is our actually the interest. But when we used Monod equation to develop some design equation for activated sludge process, what we did, we try to predict effluent concentration. We try to predict our biomass concentration, right? So yes, model always has some limitation. You can extend, and that's what actually researchers are doing. And as I mentioned, some researchers actually extended this model to Nernst Monod equation. Some people actually extended Nernst Monod Ohm equation. Okay, so there could be a lot of extension, but what we found, so if only one term can actually fit your experimental data, that means those other component or other extension may not be that critical, so they are not rate limiting. This is the one is actually rate limiting. Okay,
this is the basic of all this.
Yeah, so this is basic of all, and this is actually the beauty of Monod equation. It kind of like works.
For other water developments and applications.
Yeah,
Reference.
Huh?
Based on this information, we can develop some other
Yeah,
models.
yeah, obviously, yeah, there is always room for, you know, like you can extend the model if needed. There are a lot of things you can do, yes. Okay, so I'll stop here and thank you so much. And this is where I'm kind of finishing right now. And I think we already have another speaker here for today. I'm just going to get my, yeah. Okay, just give me a second. Let me clean the board.
Thank you, sir, for sharing your valuable insights and expertise with us. Today with us, we have an invited speaker from IIT BHU, Varanasi, Dr. Jyoti Prasad Chakravarti, sir. So I take this privilege to introduce him. Dr. Jyoti Prasad Chakravarti is an associate professor in the Department of Chemical Engineering and Technology at the Indian Institute of Technology, Varanasi. He earned his PhD and MTech degrees from the Indian Institute of Technology, Kanpur in 2011 and 2003 respectively, and completed his BE at Jadavpur University in 1999. His research expertise encompasses chemical reaction engineering, torrefaction, pyrolysis, gasification, and kinetic modeling. He has several international peer-reviewed research publications and patent to his credit. He has done many government-funded projects. Throughout his career, Dr. Chakravarti has recognized several awards, including his Best Oral Presentation Award at ChemSpark 2022, the Best Poster Award at Chemference 2008, and an International Travel Award from CSIR to attend Pyrolysis 2008 in Spain. I would like to invite Dr. Jyoti Prasad Chakravarti sir
I don't know how to hold it. This way. इसमें ये यहाँ पे कर दीजिए. Am I audible? I think without that also might be
I think that's only for the video recording.
recording.
Yeah.
Okay, okay, fine. Yeah. So now you tell me how to shuffle this arrow, right arrow. Just okay. Just can you make it full screen or it's already full screen? Already full screen.
Here it is only this.
Okay, so just 1 minute. Let me just. This is the shuffle, na?
Yeah.
No, it's not going.
From here.
उसको कर दो. हाँ, then एक arrow होता है ना, right arrow. It's going, na?
Yeah.
So this is visible to me, but it is visible to everybody.
Yeah.
Oh, so I have to.
This will visible to all and this will only.
Okay, I have to go to the first slide. Yeah.
Thank
Okay, thank you. Thank you, Professor Joy, and some other faculty who we have our student from IIT BHU, and she accompanied me for last more than 1 hour. So I thank you all for such an intensive theory, practice, experiment, and model equations, and a lot of brainstorming equations. But I fear I don't have that much equations. Okay, and so far what I understood from the invitation from Professor Joy and the other documents, that it is mostly, the discussion is based on some process, some experiments conducted at low temperatures. Okay, and I usually work on high temperature process, and that might be a little boring for you, or fresh air. I don't know. Okay, so let's quickly move on to that. The topic I am given is the waste to wealth, and waste to wealth nowadays I think all of we are familiar with a lot of Facebook, these videos and reels and shorts. We see a lot of things happening. Okay, so I will give 2 examples to start with, and to impress the point that it is a very, very important topic, not only for research but also for a general public, because my talk is going to be very much general, because I don't know the audience. Now it's good that there are some faculty members, some students, some masters, PhD students. So let me show you one picture. So what do you see? It looks like a nest. Is it a nest? You see, this is really a nest. Unfortunately, these are strands of plastic. Okay, that means the bird was forced to negotiate, adapt with this. She's not getting the normal wood straw and all these things, rice straw. She has to collect this plastic. This might be some small strands from the cement bags, sacks. Okay, she's collecting because she has to build the nest and lay the eggs. So this is an alarming situation. Another thing, 1 or 2 years back, perhaps you have read the story that one cow was having some swelling in the stomach, and she was having a lot of pain. There was dissected, and there's about 22 kilograms of plastic was taken out from the stomach, because they can't segregate while taking anything from the rubbish. So this is the way we are going towards. So on the other hand, can we think about something that this waste can be converted to something which becomes valuable for me? I sell it, I create wealth. So that is waste to wealth scenario. Okay, so I will also talk about some low temperature processes, but I will especially give you a glimpse that through some of the high temperature processes like torrefaction, pyrolysis, we can really create wealth. And some countries in Europe, they are already in moving ahead. In Canada, from where this professor is coming, and also US, and very soon I think we are going to have a commercial scale plants where we are really producing gaseous fuel, solid fuel, liquid fuel, and so forth, or maybe specialty chemicals. Okay, now this is a classification. Broadly we all understand. So one group is biodegradable, another one is non-biodegradable. So about the non-biodegradable, some people say it takes 200 years, 250 years, and it's very long time. Okay, and it creates a lot of other nuisance also. So thing is that another small example, that suppose you have a roof, and the roof you have some plastic ropes. We just fasten with these bolts and all, and we just spread the wet cloth to get it dried. Okay, after 2 or 3 summers, do you see that it is a little bit brittle? Okay, that means there is a mass loss. There is slow chemical degradation. Where does it go? So this is emission of some kind of toxic chemicals also. Okay, so these are the problems associated with this kind of non-biodegradable materials. And on the other hand, if you talk about the biodegradable material, we have about this agricultural residue, forest residue, algae, and genetically modified plants. I have a collaborator in US, he works on the hybrid poplar. Okay, hybrid poplar means you genetically modify and plant in a commercial scale so that it has a rapid growth. Okay, then you can convert it to fuel by some other process, and then the organic fraction of the municipal solid waste. Okay, so now the processing of material. Let us for a few slides talk about the biodegradable one. The way we process the material with high moisture should be different than the process through which we process the material with low moisture. It is because water has a high heat capacity. Okay, so if you want to heat the material to 100 degrees Celsius, then to another temperature, say 400, 500, to convert it to some kind of fuel, then you have to heat that water also, that a high moisture. So for this kind of like this cow dung abundantly available in our country, and cow dung cakes in the village area very frequently we will be able to see. This is the water hyacinth. Water hyacinth is a biomass which spreads very fast. It covers the wetland, and also it creates some kind of diseases. I don't know the name, but it's not very benign. Okay, and also there will be a lot of snakes also you can find, but these are rapidly growing. So these two belong to the class of those biomass or biodegradable material which has high moisture. So you have to define some process which will be able to use this biomass to convert to some kind of valuable material like anaerobic digestion. Okay. Now some biomass like this rice straw that person is collecting, these bales and all, and this wood sawdust, that we have a very large furniture industry. So they get the wood, they wood logs from there, they cut, and all these things in the furniture shop. There's a lot of sawdust is generated. So one way we can, it has little bit high moisture, but we can using solar drying we can bring the moisture level to typically below 20%. Preferred is below 10, then we can use it for other like thermochemical processes. Okay, but these are the category of biomass with low moisture through which we can go for some kind of thermochemical processes. Okay, now with the high moisture content biomass or this biodegradable material, we can have anaerobic digestion, where the main objective is to produce biogas. Okay, so biogas, it's a combination of methane, and it's about 55 to 65%, and then remaining large quantity CO2. That is one problem, and some small impurities like H2S and other things. In our country, in the context of India, that we have a lot of agricultural based countries, so we have a lot of potential, because the farmers need lot of quantities of fertilizer. So the residue from this anaerobic digestion process, that is basically a liquid slurry. That liquid slurry we can separate it out in a liquid and solid, and solid get it dried, and that can be a very good source of organic fertilizer for the farmer, which we can sell at a lower price, and the liquid can be used as a liquid fertilizer. Okay, and gas I already told about the biogas. Okay, so then fermentation, we produce the alcohols. Okay, then hydrothermal liquefaction, that is going on. We take a batch kind of reactor, then we put high pressure, and then liquid phase, the feedstock gets converted to some valuable chemicals. Okay, and also wet torrefaction, then combustion. Torrefaction is another mild process I talked about, because we have produced 1 or 2 PhD and then couple of publications we got. And combustion, please remember that so far, though carbon dioxide is is a greenhouse gas, but in any process, if you release carbon dioxide or say moisture or steam, it is not that harmful as compared to the toxic gases. So if your objective is to get the exothermic heat, then the first choice is combustion, like thermal power plants, where we are getting this electricity production. The people like NTPC, they are doing this. Okay, and with this, let us now move on to the low moisture, or mostly because of the dried biodegradable material or biomass. We can carry out torrefaction, pyrolysis, gasification, combustion. Okay. So now again combustion we will not discuss too much because people in mechanical background they carry out this combustion, and NTPC is already blending that pellets, wood pellets with the coal and carrying out this combustion. Because of that we get the exothermic heat, and with that we convert water to steam, then the low pressure to high pressure steam, the steam turbine and electricity. Okay, that's pretty standardized, and now slowly they are experimenting and increasing the percentage of pellets being mixed with the coal. Okay, and now coming to the other process where we want to get some kind of valuable material in terms of say solid, liquid or gas, three phase. Okay. Now in this context, before I start on torrefaction, let me give you some brief glimpse about the push on the renewable energy research. One is the solar energy, one is wind, then wave, tidal energy, hydropower, and then from biomass. Okay. So in our country, you know about that we have huge coastal area, then we can get good amount of wind energy. Now there are some limitations, like from solar energy you can get electricity and thermal, solar thermal, but you cannot get any liquid fuel, right? Wind energy you can get lot of electricity, that's it, but no heat. But with the biomass we have the potential that we can get, like through digestion we can get gaseous fuel, through carbonization we can get solid fuel, through pyrolysis we can get liquid fuel, through gasification you can get syngas, that is another gaseous fuel. So those kind of variability is there and flexibility is there. So people like chemical engineers, then people from the background of biotechnology can really work into this domain. So this torrefaction is basically called the mild pyrolysis. Okay. So now if I ask you a simple question, that suppose after morning, after taking shower, I am doing some kind of puja, and there is a incense stick, agarbatti, that I am hitting. Okay, that I am hitting, what will happen? If the condition is say room temperature or air is there, I slowly hitting, I reach 100, 150, 200, 250, what will happen? Can anybody tell? Will it ignite?
100 to 50 degree.
And then we are increasing the temperature. Okay, this is one question. Another question, there is no air, and I am having the same process in the furnace, 100, 200, 300, 400, 500.
Pyrolysis.
Okay, so in the first case, one point it will catch fire because the air is there. So the product will be ash and carbon dioxide, and we will get the heat. But if if air is not there, say we are maintaining some kind of helium environment or nitrogen environment in our atmosphere. So the thing is that in the range of 400 to 650 degrees Celsius, typically we carry out the pyrolysis. The solid gets converted to vapor, we cool it down, then it gets separated, we get the liquid fuel, that is called the bio oil. Okay, but in torrefaction, basically now you can say scientifically, in the scientific literature, this name is well established and accepted, but from thousands of years we have been using this process, that's called carbonization. If you see the older pictures that people want to have some kind of charcoal, what they used to do, they used to collect the dried wood branches. They make some kind of pyramidal structure, and the outside they just cover with some kind of cow dung and all, and slowly they put a light ignition source. So slow burning, after maybe few days or few weeks, it gets converted to black colored charcoal. So here we are producing this charcoal like particles in a short span of time. Okay, here temperature is of the order of 2 to 300 degrees Celsius, but condition should be inert. That's why we call it mild pyrolysis. Okay. Heating rate is important, so preferably we use 5, 10 or 15 degrees Celsius per minute heating rate.
Condition should be inert.
Yeah, must be inert.
Not even limited supply of oxygen.
That could be there. Then torrefaction. Then the calorific value might be little bit. If there is some combustion, you will get ash. That doesn't have any calorific value. Okay. So one good thing we have observed is that after the optimized torrefaction condition, whatever torrefied biomass like the uniform black colored particle you get, that has a calorific value of the order of 22 to 23 megajoules per kilograms. That is comparable to subbituminous coal. That's why probably NTPC is investing a lot of money and blending these pellets with the coal. Now on the other hand, you see coal mining has a lot of other problems. Indian coal has ash, then the sulfur content. Sulfur is corrosive and all those problems are there, and you have to go for coal washing. Coal washing, for that you need a lot of fresh water, and after that this wastewater usually The big companies have their ETP plant, effluent treatment plant. Small companies, they have the pipeline. It goes to the drain and this river, and 100 kilometers down the line you are having some arsenic problem. You don't know from where it's coming. So in this context, if we have to produce large quantities of charcoal like particles, I want to blend with coal or something like that, then torrefaction is the best process. And why best? Because unlike the Gulf countries, they have huge resource of reserve of crude oil. We have very limited supply of crude oil, and for the energy needs 80% we are importing, and we spend about $130 billion. That is a huge amount for a country like India. On the other hand, you see that the waste biomass, that is of the order of 450 to 650 million metric tons per annum that we are producing. For example, in the morning breakfast, I have a banana and banana peel, I just throw it away without ignoring its value. Okay, so after torrefaction, now the second focus is the pyrolysis. Okay, where the most important sophistication or the importance of pyrolysis process is that this aviation sector is expanding very rapidly. Now we are connecting smaller cities with bigger cities. Okay, you might have heard that term UDAN, Udan scheme, Ude Desh Ke Aam Nagarik. Okay, what is the meaning? That the distance should be covered within 1 hour period and the flight fare should be within 2,500 rupee. Okay, that means we need more and more ATF, aviation turbine fuel, but ATF is coming from the refinery where you burning the fossil fuel. On the other hand, if we sort of take the biomass, which is a very very low cost as compared to crude oil, that is about $80 per barrel or something like that, now convert it to some liquid fuel, and pyrolysis will give you some oil that is of low quality. We have to upgrade it using some catalyst or other process, hydrothermal, all these things, okay, or hydrotreating process. Then we can upgrade it to diesel range or kerosene range fuel, and kerosene range fuel is basically the ATF. So then in a flight, yeah, you can think about military or commercial aviation, that still there are some experimental runs using solar energy, but in next 20-30 years we are going to use liquid fuel. In that aspect, there is no substitute for this fuel obtained from renewable biomass. So that is the main advantage of pyrolysis process, that if you can commercialize, we can reduce a lot of dollars spent to import crude oil from abroad. Okay.
So what is the difference between torrefaction and pyrolysis?
Yeah, in torrefaction our main goal is to get the solid product, because we get very small amount of gas, small amount of liquid, and liquid has a lot of water and all, very small amount of chemicals, they don't have much value. But the yield of solid, the torrefied biomass is of the order of 70 to 50, okay, severity of the torrefaction. But in pyrolysis, even if you have the same heating rate and all, but temperature is 600 degrees Celsius, in a fixed bed reactor, the liquid yield will be about 50%. So you take 100 gram biomass, about 50 gram bio oil you are getting. Now this 50 gram bio oil, then by upgrading 40% of that can be upgraded to some kind of petrol or diesel. Okay, so cost wise you have benefit if you are going for this commercial scale. Now coming to the gasification, where it is also called partial oxidation, because suppose C plus O2. Now if you don't supply the adequate amount of oxygen, then it gets converted to carbon monoxide. Okay, so in case of biomass, it will there is hydrogen, so you will also get some kind of CO plus H2, that is the syngas. Both are combustible, and we know the famous Fischer-Tropsch process where we can convert it to the gasoline range fuels. Okay, and combustion, if you want only energy, then we can go for combustion, otherwise we will go for other processes. Okay. Now this anaerobic digestion process, this is a typical schematic diagram where as I told you that we are getting this gaseous product. Then in chemical engineering we know a lot of adsorbents which can absorb the CO2, and we can get the methane. We can compress it to convert it to CNG, and CNG buses and CNG cars and CNG autos, they are already plying in different cities. Maybe in your city I don't know, but I have seen a lot of things in Noida and Delhi, they are plying. Okay. And with this residue, we can have this liquid fertilizer and solid fertilizer, which you can give it to farmers. And that using if you just consider about this state, Uttar Pradesh, okay, once it was a very large state, now we have Uttarakhand and cut into pieces, but still we have a lot of big sugar industries like we have in Uttar Pradesh, and many plants in Maharashtra, Tamil Nadu, Okay, so there is something called a press mud, that is a sludge kind of material. Actually, you take the sugar cane, you crush it, you get the sugar cane juice. That has a lot of mud and all impurities and sand particles. So you add some sulfite process in sulfidation process, so that there you get two phase. So clear juice goes to the compressors and all these centrifuges to get the crystal sugar, and the remaining this which settles down, that is taken out, press mud. The problem is, I worked with a company in Gorakhpur. I saw that is given to the farmers for almost free. They just have to bring their trolley and take out this material. Okay, now it has some sugar content. Initially, they are getting a high yield of sugarcane harvest, but after 2-3 years, the soil structure is getting degraded, and that knowledge farmer doesn't have. The industries should provide them. On the other hand, we thought about another process where we are bringing the press mud into our lab. Now we have lot of fruit juice stalls. We collect the mixed fruit peel, because separating each and every fruit peel is not very economical, because one fellow is selling the maybe Musambi juice, then orange juice, and then pineapple juice. So all are basically high moisture, biodegradable and carbonaceous material. So fruit peel, then small amount of cow dung, because cow dung has the methanogenic bacteria, and then we add the press mud in different ratios, and we have seen that you can get an optimum value of about 80 to 100 meter cube of biogas per ton of biomass. So that is the potential of this process and the value of the byproduct. Okay, and another advantage is that this process is almost temperature heating wise it is neutral. 37 degrees Celsius you have to maintain. So for the remaining months there is no problem. Only in the month of January and February, where the temperature can go down up to say 5 or 7 degrees Celsius, we have to provide some kind of heating. Okay, but that is also still economic. Only thing is that this is a slow process. It takes about 1 month to complete the this process. Okay, this anaerobic digestion process. So similarly, we have this hydrothermal liquefaction process, where we get some kind of you see that we take the biomass, then do some kind of pre-treatment like washing, then cutting into small pieces, and then after this process, that residue we can have anaerobic digestion and other things. Through this residue separation, we can get biochar. Biochar is another important process from pyrolysis that we will talk, and I think many of the faculty members might be working with biochar, and also we can do some kind of catalytic process, okay, with this biochar. So I would like to show you these pellets. These pellets are made out of the torrefied biomass after we compress them. Okay, and once we start from the raw biomass and then see the color of this thing, it becomes basically obviously uniformly black colored and brittle material, and also it becomes hydrophobic. So making pellets is a little difficult, so we have to add some kind of binder. So on the binder, my one student did the MTech thesis part in Tata Steel R&D Jamshedpur. So we are continuing that work. We are trying to use some kind of molasses and some other material or flour from wheat. Okay, we call it atta. That mixing, so that we want to make these pellets which will not break in transportation. Okay, otherwise these are having a very high, of the order of 30 megajoule per kg calorific value. Okay. Now in this pyrolysis process, as I told you, that only the temperature range goes up, and the biomass particle size we have optimized of the order of about 0.5 to 1.5 millimeter. That is the optimum. If you go at a very fine particle, then in the reactor there could be pressure drop problem, and if you have a very bigger particle, then problem is that inside there might be not properly converted. Okay, so with an optimum particle size and this inert condition and the temperature range up to 600-650, we can convert first of all there is tar, and tar then converted to separated to the gas, and then we got the bio oil from the bottom. Okay, now here the process is that about 25 to 30% will be solid residue, that is called the biochar. So this biochar is not torrefied biomass. Okay, this biochar is because it's much higher than the torrefaction temperature. This biochar is having a very good quality, that we can have very high specific surface area, like meter square per gram. Yeah, that we can use, and we can also use it for the removal of contaminants from the wastewater, like in the RO system, water purifiers, and also soil remediation and all these applications. It can be used to hold the water. Okay, and to make the process economically profitable, we have to do one important thing, that is the gas recycle. What is this? That typically we call it NC gas, non-condensable gas. Non-condensable gas means here we have hydrogen, CO, CO2, methane, and small amount of ethane, C2H6. Okay, in this gas mixture, CO2 is non-combustible, but the other things are, they can be burnt. So they are basically recycled and burnt, and that heat is provided to the biomass to supplement the energy requirement. So basically we can have some kind of energy recycle, and the net input is reducing because we are able to use the energy, and when the reactor cools down, we take out the biochar, and whatever process is feasible, that we can do this with this biochar. Okay. In the gasification also similarly we can have with the coal or biomass or maybe a blend. Okay, and also from petroleum refinery we get pet coke, that is called petroleum coke. Okay, so that is that can be used, and different ratio also we can mix. Only thing you should remember that when we carry out pyrolysis of biomass, we get the biochar. That biochar can be used for agricultural purposes, but whenever we are now a burning topic is that we all have a smartphone. Maybe some people have 2 phones. Okay, so very soon we are going to have lot of electronic waste. Okay, you are working, there is a printer, and so so I saw people working to optimize the weight ratio of say coal and biomass, biomass and electronic waste, biomass and some other thing. Okay, and waste plastic. So those electronic waste, there will be plastic and as well as some metals which might be harmful for the soil. Okay, so if I am carrying out the pyrolysis of biomass and plastic or biomass and electronic waste, I should properly analyze the char which I am getting. We can't call it biochar because there is a mixture. Okay, and I have to do the elemental analysis and find out what is the metal content using some XRF or some other analysis. Okay, so and then in this case for gasification the temperature range is much higher. That is an important cost, input cost. So 700 to 1200 is typically the range. We have to use, we can use air, or we can also have some oxy-fuel gasification. Okay, and on gasification modeling we had 1 or 2 papers with my PhD student. Okay, so so there also we can have this very valuable syngas, and then with syngas we can have first of all we can extract the energy. So high grade energy, low grade energy, and finally whatever the energy is left, that thermal energy can be extracted and used for room heating or some kind of heating of water, heating of air for domestic purposes also, which basically we have not seen the home heating system in India, but countries like Canada or northern part of Europe, they have this centralized home heating systems where this kind of heat can be utilized, and after that we can convert these chemicals through proper catalyst and reaction and temperature pressure conditions to gasoline range fuels. Okay, now the thing is that in our country we have availability of forest residue, agricultural residue, and many other like kitchen waste also we have lot of quantities generated. So the biomass selection criteria is very important if we want to minimize the input cost and maximize the profit, right? So this criteria is very important. We have to find out that if it is high moisture, we should not go for high temperature process. Okay, if it is having high moisture, we can try for biochemical or biological processes. If it has low moisture, then obviously thermochemical processes are beneficial, like pyrolysis, torrefaction, etc. And now the thing is that this is the something called a volatile matter. Okay, are you familiar with the term volatile matter? Okay, can anybody tell me how do we calculate the volatile matter in a lab of any carbonaceous sample, biomass or biodegradable, or anybody has worked, or otherwise I will give a brief. Anybody would like to give his feedback? Okay, how do you quantify the volatile matter? So basically we take biomass in a crucible, we'll put the lid and keep it in a furnace at 925 degrees Celsius for 7 minutes. So quickly it gets converted to vapor. okay, and then from the mass loss we find out what was the volatile matter. Okay, and I also talk about the TGA process, thermogravimetry, that is very, very important for us. So if we see that the volatile matter is high, that means after condensation I will get high quantity of liquid. That you are able to understand. If the volatile matter is 80%, then 80% goes into vapor, okay, then you cool it down, you get 80% that approx quantity of bio oil. So if my objective is to get liquid fuel, I will search for the biomass which has a low moisture and high volatile matter, okay. And obviously ash is basically a collection of different metal oxides. They don't have any heating value as say so as such, so that we will search for ash biomass having low ash content, and usually the good thing is that biomass has very, very less sulfur content and very, very less nitrogen and ash content. Only a few like rice husk and all, they have high ash content, but majority of the biomass has ash content of the order of 3 to 4. On the other hand, if you take Indian coal can have up to 30-35% ash. Okay, so ash removal, ash beneficiation is another big challenge.
Low moisture means up to what percentage?
Typically below 10% is best, but below 20 is also workable. But this hyacinth and cow dung, they might have 70% moisture and 65 or very high. So you have to remove the moisture, then only you can get the carbonaceous material. So your lot of energy will be wasted in heating the material.
Okay, sir. I have another question.
Yes.
If I want to have some adsorbent
Yes.
that is having wood specific or the paste specific
Surface area, yes. Yeah.
and paste porosity it can have. So can I go with pyrolysis, or I should go with torrefied process?
Torrefied process you will not get biochar. Whatever the torrefied biomass you will get, that will have limited surface area. Suppose I am telling you, suppose from biochar for pyrolysis, if you get suppose 50 meters square per gram, there you will get 4 to 5 or 6. Because lot of chemicals are not yet released. The temperature is 2 to 300 degrees Celsius only. So lot of volatile matter is still released.
And is there any kind of sticky parameter or for pyrolysis? I see that people start from 500 to 1000. So
They can go.
yes. So is it variable for different biomass, or 500 to 600 is the optimal value for me?
Okay, okay, very good question. Pujan, note this question. Okay, so may I have one marker? It's okay, okay. Okay. Typically, the thermogravimetric analysis, it is very, very important for our study at the beginning, the biomass, coal and all these things. So here we plot the mass percentage versus temperature. Also we can plot time. So 100% means you are starting with say 5 milligram, 10 milligram of biomass. Now when the temperature reaches about 100, moisture loss, then it has 3 important components: hemicellulose, cellulose, lignin. Others are ash and extractives. So now this hemicellulose, that gets decomposed within the range of 2 to 300 degrees Celsius. So during torrefaction the hemicellulose go, but majority of the cellulose and lignin will remain there. So they will not create good quality pores. So the specific surface area will be less. Now cellulose mostly gives us the liquid fuel, that bio oil. Okay. So for pyrolysis process, I have to carry out the TGA for the unknown sample, okay, and mark this range. Say for example, it is 350 to 600, suppose. So in that range, what we do, we assume 3 temperatures covering the entire range, and there is a we have a optimization software. There we input this, and then it gives us some theoretical data points, that at these data points you have to carry out the experiments to get the final optimized value, that whether it will be 500 or 490 or 510, whatever. So in this condition, cellulose will live, and some part of lignin, that complex polymeric materials, network structure. So if the lignin also sometimes to some extent degraded, then the pores will be available. So we can have high specific surface area. But for using as an adsorbent, that is also not sufficient. We have to do some kind of pre-treatment, some kind of, people do acidic or alkaline, so that is very important. But pre-treatment, it has one challenge, is that after that the wastewater that has to be properly taken care of, recycled and all these things. So TGA tells us what is the lignin content, what is the cellulose, what is the hemicellulose, the temperature range and all these things. So with that, then we take the final decision. Okay, and obviously the total is 100. So whatever this fixed carbon is, basically we cannot compute through some instrument, so we subtract from 100 whatever is left out, that is the fixed carbon. Okay, like you can say mostly it's the biochar. And if you look at this plot, this is, suppose this is 1000 degrees, okay, and you see that here we have a parallel line, okay, parallel line through to the x-axis. That means no more mass loss is taking place. That means here it is, suppose this is 10 or 20. The 20 will be the combination of fixed carbon and the ash, because whatever material you have started, it has some ash initially. So sometimes people tell that it is only fixed carbon, but it is basically fixed carbon plus the ash content. Okay, so so this is called the proximate analysis. So any student who wants to start with some kind of similar process, that I want to produce gas or solid or liquid, you must carry out the proximate analysis. For that, you just need this furnace and or oven to find out the moisture content, and furnace can give you the ash. Okay, ash, how do you do this? For biomass 575 degrees Celsius, 4 hours, you just keep the crucible open, so that if we allow the combustion, whatever ash is remaining, that is the percentage we find out. And for volatile matter, it should be closed, because inert condition and pyrolysis will take place. Okay, now I will have a little bit discussion on the waste plastic. So plastic pollution, you see that there are some plastic, means in the domain of waste plastic, which are having only hydrocarbons, carbon and hydrogen. Okay, they are still manageable, because if we convert them through pyrolysis to liquid fuel or gaseous fuel, we will get some kind of hydrocarbons only. Problem will come with some kind of, say this is blue colored material, right? So we can assume that probably some kind of polyvinyl chloride is there, PVC pipes, we make a lot of pipes and those buckets and all these things, the hard things. So this PVC, when we carry out experiments, then there might be the toxic chlorine gas is emitted. That is very, very dangerous for our lungs, our human body. So in fact I visited one plant of Birla Group where they produce chlorine, Eastern UP. And you have to take a lot of precautions before you are allowed to enter the factory. Okay, so this waste plastic, as I gave you the example of the cow and the bird, and also microplastic, that the micron size, and those microplastics are entering the food cycle. Now I think everybody know that fish we are consuming and getting. Even recently, the microplastic was obtained or was found in the mother's milk also. So now think that the next generation we are passing out this problem. It is a serious problem globally, right? So some examples of how we use different, like that polyethylene terephthalate, high density polyethylene, PVC and all these things, and typically how we use it. In fact, I saw a video, it's very, very interesting. One lady, probably in Bangalore, what she was doing, that milk pouch, half a liter, that Amul blue colored, red colored, and what do? We just use the scissor and cut the corners. That small part is left out. That cannot be recollected. It's very small in size. Okay, so she thought that, okay, I'll just make a small hole with the scissor, and that will be there with the body, so that someone who is collecting that pouch, that will be a single cut, but the pouch will be there. So that these are the like every institute has this innovation cell. Like people, I don't know whether if the student is there, BTech, anybody BTech, they're busy, coding and all this, coding, and Python is not a snake. Python programming language. Okay, anyway, so so any idea comes to your mind, I as a faculty should be flexible enough to receive your ideas and let us discuss. So these small things can bring a lot of impact for the society as a whole. So food containers and all these things have the plastics, and please remember that there is this kind of recycle structure, and between there is a number 1 to 7. Okay, so these 1 to 7, what they represent and all these things, and this word is very important for us, goes to landfill. Now India, I think 40 to 60% is doesn't go to landfill. That's a huge burden. Where does it go? Anywhere and everywhere. Okay, so even the great trains like Vande Bharat or Rajdhani, we get the announcement that help us to maintain the cleanliness. That they request us to be careful. Okay, so these whether they are recyclable or not, that general awareness as a citizen I should have. Okay, and then and I heard, I don't know, Professor will be able to tell, Canada is the best country to have segregation at source. They have outside their flat that 3 or 4 type of containers, that biodegradable, non-biodegradable, wet, dry, then metallic. This is a very nice thing. Like BHU, about 30,000 students are there. So they are, suppose you have some shaving part, and that is now not working, you are throwing, and some banana peel is also there. So that one gate is open. Okay, so cow and all these things, so there might be big problem in their mouth, cow, dog and all these things. So these things, if we are a little bit mindful, we can make a better world. Okay, so this is the plastic pyrolysis where collection. In fact, I couldn't verify, but someone told me that the largest plastic producer, basically Reliance, and they are also big player in the recycling. Okay, so that we don't know, but so rag pickers and also they collect and get some kind of rupees per kilogram, and that is collected and cleaned, and then you see different types of, depending upon the that mass transfer, like this the distillation column temperatures, we will be able to separate them, like this atmospheric, then vacuum distillation tower, we can separate them to light oils, then kerosene, then diesel, and ultimately some kind of carbon char. This carbon char, usually we should not throw it to the land for all soil remediation purposes, but we can have some kind of the tire of the car, where this petroleum coke is used. Okay, so these kind of things can be produced, or as a pellet, for to be used as gasification process. Okay, so the characterization of sample is very important, because as a student, if you are starting your research, you should doubt your own data, you should doubt your own supervisor. Okay, because whatever I am telling, this from my experience, I may be wrong. Okay, so science is such a beautiful subject that you should verify for your own. I am giving a rough idea that this is a high moisture, but what is the moisture content? So you just keep it in the oven for, say for example, 2 hours, 105 degrees Celsius, and you see the mass loss is the moisture content. And please remember that you should repeat at least twice. The 3 experiments are best, and then you will be able to plot the error bar. Then there is an important thing is called the ultimate analysis. This is a little costly instrument where the entire sample is burned to a very high temperature of 1000 degrees Celsius, and then we get carbon dioxide from there. We get the quantification of carbon, hydrogen, nitrogen, sulfur, and from 100 subtracting we get the oxygen content. Okay, so we get that, okay, how much carbon it does have, how much sulfur. If sulfur is more, then we have to have a lot of processing that is required. Otherwise it's very corrosive gas. Then TGA, I just showed you one example. Now TGA is giving me at the first hand that the temperature range at which I should carry out my pyrolysis. Fine. Now we can also do 2 more important things by this TGA data. Okay, one is carrying out the proximate analysis using oxygen as well as nitrogen. Suppose if it is in nitrogen atmosphere, then we can have this, this is moisture, this is the, say, biochar. In between you subtract, that will be the volatile matter. Now you switch the gas from nitrogen to air. Same instrument TGA. What will happen? Combustion will take place. So what will be left out? Only the ash content. So you have moisture, volatile matter and ash. Subtract from 100, you get the fixed carbon. Okay, but in India we basically, this is 30 lakhs instruments, so we get a little bit fearful that, okay, I will open and all these things. So we have dedicated staff for that. But in just having 2 cylinders, we can have very accurate data for proximate. Okay. Second one is kinetics. Kinetics, what is kinetics? Kinetics gives me the speed at which I can complete the reaction. That there is an 8-hour cycle for any staff. So if it's 24 hours, so three kind of cycles, shifts. So I have to try to shift the complete the experiment within 8 hours. So how will I be able to do? So how fast the reaction will take place, that will be governed by that the kinetic triplet, we call it, that is typically if it follows the Arrhenius law, we say that it is k equal to k naught into e to the power minus e by RT. Okay, so basically that n e then k naught pre-exponential factor, these are called the kinetic triplets. Okay, order of reaction, then activation energy, that gives the barrier, okay, that you have to overcome to have the proper collision and formation of products, and this is the k naught is the pre-exponential factor. So those things can be found out using some standard models are there, which you have to feed the data of TGA, and that gives me the value of E and all these things, and from there I can also calculate the thermodynamic parameters like the change in enthalpy, entropy, and gives free energies. Okay, scanning electron microscope gives me the surface morphology, that if you do for torrefaction and pyrolysis, you will see in pyrolysis there are a lot of more porous structures, because the volatile matter mostly they have escaped, so pores are formed and you will have higher surface area. Then functional groups like OH, CO and all these things can be found out using this FTIR analysis. So that these are basically very useful, and these things are not very costly. So if you need help, you can approach me. Okay, but only those like CHNS is little costly. Bio-oil analysis is little costly. We send to JNU and or IT Bombay. Okay, because sometimes instruments are not working. Now this is my favorite plot. This is called the Van Crevelen diagram, where you see starting from biomass, we are going through torrefaction or pyrolysis. What we are doing, we are reducing the O/C ratio, going this way. Okay, so in this process, this arrow shows that increased heating value. So biomass typically has 12 to 16 megajoule per kg is the heating value or calorific value, as is standard case most of the biomass. Okay, now through torrefaction I am able to get to sub bituminous coal, and the best quality coal, that is the anthracite, that has of the order of 35 plus, and petrol, diesel, they have 40 to 45 megajoule per kg. So this scale, if you remember, you can find out that how far can I go using my biomass. Okay, and if it is higher, then definitely you can blend it with coal and use in this pulverized coal fired furnaces. So this Van Crevelen diagram basically dictates that starting from biomass for my biochar or torrefied biomass, how far I am able to go down that in the O/C ratio. The only problem is that along with O/C, you see the problem is this reduction of H/C ratio also. Okay, so in terms of fuel properties, H/C should be high. Just think about the fuel which is very easy to burn, easily it will catch fire because H/C is very high. Okay, now you take some liquid fuel, liquid at room temperature like this, heptane, normal heptane, C7H16. Okay, so what will be the value? 16 by 7 atomic scale. So it will be little bit more than 2, but much less than 4. So liquid, little difficult to burn. So coal even low. So in this case we are having some problem. So that if you do the same thing for bio oil, you will see the same problem. So we carry out some kind of experiment that is called the hydro deoxygenation, where the oxygen of the bio oil is taken out and replaced by hydrogen to improve the H/C ratio. That means burning characteristics got improved. Now the question is, in biomass, from where do we get the oxygen? Do you have any idea? Anybody, any MTech or PhD student? From where do you get so much oxygen in biomass? That basically is a problem for the fuel. Water. Okay. Moisture. Okay. If we remove the moisture, then also the problem is there. Because this is a product of the process photosynthesis, where we are absorbing carbon dioxide in the chlorophyll. So that oxygen, we are getting glucose, C6H12O6, right? So that is the difference in formation of biomass as compared to crude oil or coal. In crude petroleum and coal, we have very limited oxygen, but maybe high sulfur or nitrogen. That is another problem. So biomass has many other advantages like very low sulfur, very low nitrogen. Only oxygen, moisture might be high in some of the biomass, and oxygen is little higher. Okay, and water also. So TGA, as I discussed, the C. Now, I think to complete your question on the temperature range selection. So you see this blue colored, this is HDP, okay, single line, only the volatile matter, because plastic has very nominal char kind of content. Almost you get 98-97% vapor. So you will get very good quality, a very good quantity also liquid fuel from the plastic pyrolysis. So if you mix plastic with biomass, your overall oil yield is going to improve. Okay, and at the same time you are able to utilize lot of waste biomass. This utilization is very important from the point of view of management, because once I went to a sugar mill, there they send the sugarcane bagasse, that is another biomass, to the paper industry. One particular year there was no demand, so the one mountain quantity of bagasse was left out, and there was rainfall. So there's some biological degradation, and rats, they will spread the disease. And most importantly for an industry, space is very important. So even if you convert any type of liquid or gaseous product out of this biomass, you see that since biomass has a very low energy density, after converting it to oil, you can fill it in a drum, but the same amount will be equivalent to 1 truckload of biomass. So that's why for the biomass to biofuels sector or waste to wealth, this domain, we talk about the segregated bioil production. Like there is a huge paddy field, there you set up a small plant, then from there bioil you can easily carry through a small truck to the main refinery, and there is a concept of biorefinery. There all the processing units are there, you just plug into this low quality bio oil to some say hydrotreating unit that is already in operation. Finally you will get higher yield of petrol, diesel, gasoline and all these things. Okay, so this TGA gives us very clear idea that what are the components which are getting degraded and what is the temperature range for that. So some examples I should give you, because otherwise it will be a dry presentation. This guy, I recently saw this this video. It was a TED talk. So Kumbhi Kagaz Private Limited. So initially he showed a picture of python, that is the ajgar, that's snake, and he went to an expert and he told that okay, python, you like python snake, very good, then there is a python and all these things. He got trained. Then he was asked to take the snake to release it in some wetland. In the wetland, there was some mud and a little bit of water, and hyacinth was huge quantity. So as soon as he released that snake, immediately a heap of water hyacinth came across, snake got vanished, and then this the stem, which is basically very lightweight and it's basically fluffy material, outside green, inside white, that is the stand. And then there's a flower, right, bluish colored flower. So a lot of moisture. Then this idea came to his mind that can I convert it to paper? Then he did some research, and so this A4 sheet needs about 20 liters of fresh water. But if he converts that water hyacinth to paper, you need only 2 liters of water. So in India there are many places where there is huge water scarcity. In fact, I have a relative and she is doing BTech from VIT Bhopal. And due to water scarcity, they are giving this end semester exam in middle of April and go back home. You come in July and then you give the exam. Sorry, classes will be over by April first week. You go back home, and then in July you will, because there is water scarcity. Bangalore already almost announced that there is no, and Johannesburg, I think there is no groundwater. So this kind of innovative. We don't ask whether he's a chemistry graduate or mechanical graduate, but these ideas. So he is able to successfully produce paper from water hyacinth and commercialize. Okay, and recently a UK based company, they have produced this is the poster boy, hydrogen. Now everybody is and the government giving a lot of fund. So plastic to hydrogen, obviously plastic to pyrolysis, if you carry out, you will get a lot of gases. You will have more of methane and hydrogen, because plastic has very little oxygen. So you have to just tune the reaction by suitable catalyst to get more of hydrogen. Okay, so that they have run for say long hours, maybe close to 1 week, and with some with success. Then now they are going for the scale up. Okay, now for a non-technical person also, you can do a lot of wealth creation from the waste. This is bamboo. Okay, bamboo. I don't know about other states, but in northeast we get bamboo in very abundant quantities. Okay, so mostly they are used for paper industry and also this kind of things, where you will see that this kind of chairs and all these things people are using, and they are long lasting and biodegradable, and a lot of demand for this kind of utensils are also there. Now you all do this puja, Om Ganesha. This is coconut shell. Okay, this is a very low moisture biomass, very high. It has very high potential to be converted to particularly you can target the adsorbent. Okay, very hard and good quality adsorbent we can produce, and carbon based catalyst also. Now coming to this coconut coir. Okay, many Indian companies, they are producing this from the that matured coconut, and if you take out the coconut shell, in between that fibers, there is a coconut coir, and they are making this kind of these carpets and exporting. Like there is a place called Bhadohi, 30 km away from Varanasi, that works only on export basis. Like suppose 3 feet by 10 feet or 7 feet, that costs about 25,000 rupees, which we can't afford. So they are there is a student, and her husband is basically has this company. He showed me that it's quite heavy. This we will not see in normal shopping malls. Those are quite light, and he told me, you just try for yourself. You lift. It's very heavy, and it can withstand water and all those things for several years, and they are all exporting. So this is another, and ropes, coconut ropes that we all have seen, and maybe we have used. So how much time do we have? Because this is challenges, the theoretical discussion, I can go quickly. Okay, so one important challenge is that one is the education and awareness. Okay, because if you go to a village and tell a person that okay, I can convert from dried branches to diesel, he will laugh. Okay, so don't get disheartened. Factually we can do, because it is carbonaceous material, right? Now then collection. Those things is a still an issue, and people are working, and with the help of a lot of advanced technology, they are doing this. Hopefully we will have this commercial scale production very soon. So so mixed waste is a problem, because we throw anything into the dustbin without caring whether it is meant for biodegradable or non-biodegradable. So implementing strict sorting policies, okay, that and awareness is very important, particularly for our country, and we don't have adequate infrastructure for those incinerators, okay, because many things is not going to the landfill, it's other way around. So it is disturbing the soil, water supply, and many other problems in the municipality. Okay, and initial cost, so maybe some government subsidy which is there for solar rooftop. People are getting a lot of subsidy. For this kind of subsidy is we request the government to give this kind of subsidy, so that we can start the commercial scale. And technology gaps, something we are doing at lab scale, we are successful. Then to take it to the the pilot scale, suppose I need about 3 to 5 crores of rupees, then maybe I am not getting that fund. It's difficult, so I have to approach companies, and some government support might also be required. Okay. Market challenges, that there are big players like Ambani, Adani, and all, so they might purchase technology from abroad and install here. Okay, so indigenous technology, to take it to the commercial level, we need some tax holiday like for other sectors. But I will give you one example of Govardhan scheme, where one unemployed youth can have this plant where they are converting this cow dung and other biomaterial to biogas. Then if you can't purify it, don't worry, you just compress it. Is called CBG, compressed biogas, and there is a contract between that person and the oil marketing companies, that IOCL, BPCL, HPCL. They will come with their truck, okay, tanker. They will fill the gas, whatever based on volume and all. They will pay you within 1 month. You will get the payment, and the remaining solid residue you can sell it as a fertilizer. So those kind of schemes are giving a lot of encouragement to the local youth, and awareness participation for that. Maybe we should approach panchayats, but I talked to one panchayat, they told that also we don't have that much fund for this thing, because panchayat gets a very small amount of fund that is utilized for construction of roads, then school buildings and medical facility, this bare minimum. Other extra things they don't get, maybe they get before the election, but I don't know. Little bit of, and regulatory policies also we should debate and all these things. Or concerns means what are the gases without any analysis, I should not say it is a clean gas. Okay, so for that we should be like this. There was NASA took a photograph from the space, and they saw that this coal industries are creating a lot of SOx emission, particularly in India. So that scrubber technology they insisted us to install, and companies are approaching the government give us some kind of 2-3 years desulfurization. Right, you are very true. So those kind of things are. So some of the global best practices we should also keep focused, that circular economy approach, like my waste material will be your raw material, so that ultimately we are sharing our utilities, then the water and other things, and ultimately what we get as a byproduct, that is, you can say, the happiness. Okay, then the industrial symbiosis, that we talk to each other and try to solve each other's problem. Smart waste management, and there is a Kamikatsu, that's a city in Japan, there's a zero waste town, there's and Sweden is also working great, and as I told you that Canada is leading, okay, the way. So role of education and mindset, awareness, basically it is the mindset. Okay, the mindset is that if I can do this, then probably I will be able to do. If I think that I cannot, then I do not have the first step. Okay, so that please remember that so, and whenever you are within an institute, you are handling a lot of fertile brains who are very young. So those brains should be given the basic minimum platform that you think, and whatever idea coming up, you might fail. Okay, so put it on a paper and then discuss with your professor. Okay, some problems need very small amount of funding, but initially we think that there is no facility, so I will not do. Please, students, please take the lead, because faculty members, we have our families and some other responsibilities, so you have little extra time. Okay, then empowering communities, and like the coal and cow dung blending, and then make the cow dung cake, that will give you large little higher heating value. So that concept, if you share to those people who stay nearby to the coal mines, they will get a lot of benefit out of that, because they are collecting coal maybe free of cost, just from that this thing. So with that, let me thank Professor Joy and your whole community to come and share. And any question? This side, they're sleeping or hungry, I don't know, but no question.
Probably
so,
hungry.
yeah. Please, any question? Any question about BHU? Though I am not the perfect person, but it is 106-year-old university. We have a lot of greenery inside the campus, and any other question from anybody else? Question about your future plan? Please, tomorrow the professor has some session. Yeah.
As we can see, Varanasi consists of a lot of temples.
Yes.
They are also producing lots of waste and all.
Yeah, those bel patta, then flowers.
So anything which are something on which you are working about it?
I have not. I heard somebody is collecting, but what they are doing, I don't know. But I tried one thing and I failed. That is the BRC, Bhava Ramkrishna Center. Okay.
They are making Essen sticks.
Yeah,
Essen sticks. So the BRC developed technology for biogas production from using the hostel mess kitchen waste type, using anaerobic digestion, because BHU has this agriculture. They have this lot of animals and all these things. So you get a lot of cow dung also. So cow dung is available. So that technology, and for 2 years I was a visiting professor of BHU economics. And I told students, and they went to the central office where you have all the files. The file is missing. Yeah. So that is, I failed successfully. I tried my level best. I failed. So the thing is that so it is. I love to fail. Because some students, after that he was mechanical background. Then he got a job in Philippines, and from there he called me, sir, I am easily able to understand many things because you taught. Because in economics they know, they study the profit loss and all these things, but they don't know that biomass selection criteria, isn't it? So if you club these two things, you really can visualize that, okay, I can set up at least a 10 kg per hour plant or 1 ton per day plant. That confidence you have once you have this knowledge, So this kind of mindset you should have, and maybe you should continue. If you are doing MTech, then in PhD you continue, PhD then postdoc, okay? Because research gives us some extra thing which that happiness will not come with salary, isn't it? Okay, so
Can you name the software on which we can find out the optimum?
it is a design expert. Design expert. RSM, yeah, RSM. You just search for my Google Scholar profile, then you get a lot of publications. And yesterday Pujan's paper got accepted, so he is the champion. Being an MTech student, he published one paper, so I am really happy that he is a very honest and hardworking guy. Got a job also, And Ankita is now she will take the lead in the sustainable aviation fuel project that I told you, that we can also produce kerosene grade fuel. So setup is already installed. The chiller is some problem, having some problem. As soon as it is resolved, we will work on the catalyst, and we have tie up with IIT Kharagpur, where this turbine is there. So testing of the fuel will be carried out in IIT Kharagpur. So I plan to do something good slowly, and okay. So if there are no further questions, can I sit?
Now I would like to request Dr. Joyabrata to present a memento to Dr. Rupika as a mark of gratitude.
मैडम को भी बुला लो, alumni है, so मैडम का है नहीं है?
Thank you, sir. Now I kindly request everyone to join for the lunch.
Hungry person.